Well, good morning, everybody. Welcome to day 5 of 6 days. Buenos dias, buongiorno. Here, day 5 of 6 days. Seems like flying by. And we heard a lot of ground in the first 4 days. And so you're on. In fact, I'm so glad you're here on time. So what we're doing, where we're at is we're in, just to refresh your memory, we're in carbohydrate metabolism, and we're on the last major pathway of carbohydrate metabolism, and that is the all-important hexosmall phosphate here. So now this pathway, let me say something about the pathway. It is the most important one, because you see we talked about the issue of thiamine yesterday, right? And now you're also going to see why it's so important for boards, because now we're going to go back about this all-important enzyme, glucose phosphate hydrogenase. So again, what was the function of the hexosmall phosphate shunt? Two main functions. One is to generate reducing power in the form of ADPA, and the other is to make ribose, to make nucleotides and to make DNA. And all right, now the idea is, so what is the function of the electron ADPH generated by this all-important enzyme G6PD? What is the body going to do with that electron ADPH? You don't make ADP from it, but what does the body do with that? And the answer is, what the body does with electron depends on 3 things. Location. What's the other one again? Location. Oh yeah, it's location. It depends on those 3 things here. Location. So what we're going to do right now is this part of the pathway right here at the beginning of the pathway, take that part of the pathway and move into different locations. And in these different locations, ask, what's the function of what happens to that electron in ADPH? We're going to take this part of the pathway and now move it into the liver on page, now what you see here on page 212, this is the top of the figure and the bottom of the figure. Now let me say something about page 212. The day before you take the national exams, you might want to revisit page 212, because on page 212 it's such a high yield figure. Why? Because integration of chemistry with pathology, pharmacology, physiology, immunology, microbiology, they're all those pathologies are on this single integrated together. So that's why it's so very high yield. So in my opinion, you got to know this page. So much high yield stuff here. All right, so here it is. This part of the pathway, we're moving it into the liver, and here's the G6PD. It makes pentoses. It makes pentoses, and it makes click on here. Okay. All right, not sure what's going on here. Anyway, it generates enzyme G6PD, then makes NADPH. Now, in the liver, what does what happens to the electron in NADPH? Well, the reductant NADPH is then being used to put the electron to carbon and other molecules, to make fatty acids, to make cholesterol, to make nucleotides. It's used for biosynthetic reactions in the liver. Some of things like cholesterol synthesis in the neck, and so don't worry about the details yet, but just know it's being used to put electron back into carbon here, because it's a high electron. So now we're going to take this part of the pathway right here and move it into another location, and that is in the neutrophil, and now integrate biochemistry with immunology. So here is enzyme G6PD, and here is the NADPH. So now in the neutrophil, from Dr. Ali's lectures, what's that electron being used for? Well, as we all know, it's being used to carry out the respiratory burst in neutrophils and phagocytic cells. So a phagocytic cell engulfs a microbe. Remember from Dr. Ali's lectures, you turn on the respiratory burst, and this major enzyme called NADPH oxidase will then take the electron from NADPH and puts it on oxygen to make superoxide. the beginning of the respiratory burst. And then superoxide then converts to hydrogen peroxide. Also, enzyme myeloperoxidase can then generate hypochlorite, and those free radicals then are being used to kill incoming microbes. So when a neutrophil engulfs a microbe, you turn on the respiratory burst, and free radicals then take electrons from the bacteria and kill the bacteria. Is the normal situation that goes on in the neutrophil here. All right, now the idea is this. Now kind of grade. So biochemistry, immunology, pathology, here comes the pathology. So now in G6PD deficiency, understand what goes on in the neutrophil, all you got to draw arrow up or down. So with G6PD, the arrow is down. What about the ability to make NADPH? Down. What about the ability to make superoxide? Down. What about the ability to make hydrogen peroxide? Down. What about the ability to kill microbes? Down. Therefore, you would predict, based on that, that one of the hallmarks of G6PD deficient immunodeficiency. These people are susceptible to infections because they carry out the respiratory burst adequately. So, all right. Now, the National Board loves this because you already had this with Dr. Ali. That because another disease out there in pathology that sounds very similar to GXP, in that the people are very susceptible to infections. What's the name of that other disease? That's right. That's chronic granulomatous disease. Now, in chronic granulomatous disease, what is the primary biochemical defect? What is the defect in CGD? What's the name of the? What's the problem? What's the primary defect? That's right. It's the enzyme. NADPH is defective in chronic granulomatous disease. Substrate NADPH is fine, but the enzyme that uses it, the oxidase enzyme, is defective. So therefore, it cannot make superoxide and then the other free radicals there. So again, now the national earlier, when you have two or more diseases that sound very similar, how would you differential diagnosis of those two diseases? Here, so here it is. Deficiency, they are deficient. In CGD, they are immunodeficient. So how would you do the differential diagnosis? Let me see your answers here. How would you do? What makes sense to you? How would you do the differential diagnosis? Well, one of them is bodies, and the other is hemolytic anemia. Now, let me ask you this. I'm looking for a yes or no answer. In CGD, do you have Heinz bodies in chronic granulomatous disease? Yes or no? And no, hemolytic anemia in CGD, yes or no? And the answer is no in both cases. Why? Because of three things: location. Heinz bodies and anemia have to do with erythrocytes. CGD has to do with phagocytic cells. So they're not interrelated there. So in CGD, there's no Heinz bodies, there's no hemolytic anemia because there's nothing wrong with your erythrocytes in CGD. So that's the idea. That's how you do the differential diagnosis, because in CGD they are immunodeficient, but there's no hemolytic anemia because of location. There's nothing wrong with the erythrocyte. All right, now just one more item here is related to the pathoimmunology, and that is now remember from Dr. Ali's lectures, you can also have myeloperoxidase deficiency. Remember, myeloperoxidase is the enzyme in the respiratory burst makes hypochlorite. Now that enzyme can be defective, and so now how would you know in pathology as well as immunology? How would you do the differential diagnosis to determine deficient, whether it's due to NADPH oxidase deficiency, or is that due to which is CGD, or is that due to myeloperoxidase deficiency? How would you do differential diagnosis? That's right. From Dr. Ali's lectures, the way you do it is by the MBT assay. The MBT So if it's positive, that means that the enzyme is working, NADPH oxidase is working. But if it's negative, that means the enzyme is nothing. So again, the MBT test is a major test for distinguishing the pathoimmunology of what's going on in phagocytic cells. Coming up here, we're going to take that 6P in the product NADPH and now move it into yet another location. And that is now is the erythrocyte, shown at the bottom of the slide, of the figure here in your book, on page 212. So here's the enzyme G6PD, and here like NADPH. All right, so now what does the erythrocyte do with the electron in NADPH? Not being used to make AD, and used for vascular, unlike the liver, makes nothing. So what is the electron being used for? We all know, and we touched on it the other day, is being used for oxidation mechanisms to get rid of free radicals that are accumulating in the erythrocyte here. All right, now before we talk about how do you do the detection of free radicals in the erythrocyte, we first got to explain where are the free radicals in the site that you have to detoxify them. Now, because again, as we mentioned the other day, most of the oxygen that you breathe in right now is in your erythrocytes. So how do the free radicals, how are they being generated? Now, we did touch on this the other day, that when oxygen goes through the bilayer, so in front of you on this one slide, if it's a single erythrocyte, as we talked about earlier, when oxygen goes through that bilayer, oxygen, because it's an oxidant, can take electrons from carbon-carbon double bonds and spontaneously make superoxide and hydrogen peroxide. So yes, and we talked about that the other day. So yes, that does happen, but it's out. That's not the major way in which free radicals are rated in erythrocytes. Yes, it is a way, which we talked about, but not the major way, which we did not talk about, because now we're going to talk about the major way in which free radicals are generated in erythrocytes. And they occur in erythrocytes. So what is the major way? All right, now again, as I mentioned, suppose this figure in front of you is one single erythrocyte. So in that erythrocyte, you have hemoglobin. And hemoglobin is it has 4 subunits. And so in parentheses, you see here, that represents hemoglobin, in which one of the heme pockets there is shown in parentheses. And in that heme pocket, there's one of 4. So in the heme pocket, here we have iron in the +2 state. Here, okay, nothing strange here. So then what happens is then in pulmonary physiology, when these erythrocytes get pumped to your lungs, the oxygen tension of the atmosphere is very high, and the oxygen is now forced through the bilayer and then comes into the erythrocyte cytoplasm and then goes on to the heme pocket of hemoglobin. So now here we have the oxygen right there in the heme pocket of hemoglobin. Here, nothing strange, nothing unusual. So then what happens? Then those erythrocytes get pumped from the lung area. They start traveling through your arteries. As they travel through your arteries, the oxygen tension is dropping. And as the oxygen tension continues to drop, then the BPG will then kick the oxygen off at the lower oxygen tension. Again, nothing new, stuff we talked earlier here. But now, it is the something that is new that we did not talk about. Now, what happens here is when those erythrocytes are traveling through your arteries and mine, carrying that oxygen. Now, before the BPG kicks the oxygen off the hemoglobin, just use your imagination. Just think about this here. That oxygen atom has never been in your body before. And so now it's sitting in the heme pocket of hemoglobin, traveling through the arteries. And because that oxygen's never been in your body, it's looking around. It's looking around all the things in your vasculature. It's looking around all the things in the vasculature. And it sees the albumin circulating by and says, "Hey, albumin, how you doing?" Then the oxygen continues to look around, and the oxygen then sees the immunoglobulins come circulating by. And the oxygen says to the immunoglobulin, "Hey, immunoglobulin, hey, how you doing? Hey, seen the microbes yet? Oh, you haven't? Oh, don't worry, you will soon. Oh, oxygen's having a wonderful time traveling arteries of somebody. And then eventually, as oxygen starts to look around, eventually, the oxygen starts to notice something. It starts to notice something. It starts to notice the iron right in front of it in that heme pocket. So what happens is when oxygen looks at that iron, it starts to notice that the iron has an electron associated with it. Starts to notice that electron. Notice that electron on the iron, and starts to notice the electron on the iron. And the oxygen starts to lust after that electron. And then the oxygen, because an oxygen, wants to know the electron in the biblical sense. So then what happens? Then the oxygen says to the electron, hey, just like Michael Jackson, hey, little fella, you want to take a walk with me? That's exactly what happens. Oxygen walks away with the electron, making methemoglobin, and the oxygen says to the little fella, hey, little fella, you know other little fellas? We can go to Neverland, we can watch a movie, I can make some popcorn for us, tuck you in bed, and things along that line. Oh yeah, that's exactly what happens. So now the tune of about 3% every day in me and you, this is how most of the free radicals are generated in your body, in the sites here. It's simply oxygen is an oxidant, walks away with the electron from the iron, and then by doing so, it makes two products, and now both these two products here are bad news here, because if that's all there was, after a week to 2 weeks, all of your hemoglobin would be methemoglobin, you wouldn't be able to carry oxygen anymore, and you would be dead. But not only that, all that superoxide damaging looking for more electrons, damaging membranes and things, and you would be dead for that. So now the body says, I want to live. And if you want to live, you got to do something about this. So how does the body deal with those two products? Well, first of all, let's tackle methemoglobin. How does the body deal with this? Matter of fact, we talked about it the other day. What happens is there's a medium yield enzyme, not high yield, but not low yield either. But this enzyme is called methemoglobin reductase. That's going to put the electron back into the iron plus 3 using the electron from NADH. So remember in glycolysis, we generate NADH, and erythrocytes is being used to put the electron back into iron plus 3 to restore iron plus 2 in the hemoglobin. So you and I can carry oxygen here. All right, now that enzyme, again, medium yield enzyme. Why? Because there's pathology associated with that. In this world of almost 8 billion people, there are some people in this world who have a partial enzyme defect, because the gene has a missense mutation for the enzyme, and as a result, not a complete enzyme defect, they'd be dead. It's a partial enzyme defect where the methemoglobin levels are like 30% compared to 3%. And as we talked about the other day, they look blue. They can have a tinge of blue like you see here, or they can have a much darker blue color. Again, they're cyanotic because of the methemoglobin here. All right, now when I talked about this the other day, I said to you, somewhere along the line, when we talk about this disease, which is methemoglobinemia, I'm going to ask you to bring in the pharmacology. So here it comes. In pharmacology, now's the time to answer. What is the name of the drug commonly used to treat methemoglobinemia? What is the name of the drug? What's the name of the drug? Anybody know the name of the drug? Not thiosulfate. There it is, Anthony. That's right, Anthony, and Kevin and Yash. That is. There it is. That's methylene blue. Methylene blue. All right. So now, I'm going to ask you. So why do you give methylene blue? Because the pharmacology person tells you to, or do you know why? Why do you give methylene blue? Methylene blue to treat methemoglobinemia? Anybody know? Higher affinity? No. Here it comes. And the reason is because Kevin, look what you wrote. You wrote it reduces ferrous. No, it's the other way around. It reduces ferric to ferrous. So in other words, methylene blue is a reductant, a reducing agent, electron donor. It's going to take ferric iron in methemoglobin, iron in +3 state, and give the electron back to it to form ferrous iron, iron in a +2 state here. So in other words, it's just substituting for the enzyme here. So, and it's a harmless molecule, methylene blue here. In other words, methylene blue is just the opposite of amyl nitrite in pharmacology that we talked about yesterday. Remember, amyl nitrite is an oxidizing agent. It removes electrons from hemoglobin to make methemoglobin for this issue of cyanide poisoning. So nitrite is an oxidizing agent. Methylene blue is the reverse. It's a reducing agent, electron donor to remake the hemoglobin from methemoglobin. So there it is. That's the why. That's the why you give methylene blue. Okay, so that takes care of the methemoglobin. So now, how does the body deal with the other product? And that's superoxide. Well, what happens there is an enzyme that'll take superoxide and convert it to hydrogen peroxide. And if I were to ask you the name of the enzyme, you're all going to say superoxide dismutase. Now, what happens here is a lot of students think, just based on the name, that superoxide is more deadly than hydrogen peroxide, just because they call it superoxide more deadly than hydrogen peroxide. No, it's just the reverse. Hydrogen peroxide is 100 times more potent as a free radical than superoxide. You know this. If, for example, I were to cut myself and I don't treat it properly, and it gets infected with bacteria or whatever, and you get all this pus and so on, what do? I go to my closet and get a bottle of dilute, and I want to stress dilute hydrogen peroxide, 3% hydrogen peroxide. I take a little swab of the dilute hydrogen peroxide, put it on the wound, and then the hydrogen peroxide will then take electrons from bacteria and kill the bacteria, and the wound heals. If I did that with concentrated hydrogen peroxide, yes, the bacteria would be killed, but my arm would dissolve away. Hydrogen peroxide is very, very potent free radical here. So now the question is why? Why are you taking superoxide and making something more deadly? And the reason is because by interconverting two superoxides to hydrogen peroxide now has 2 electrons, and it wants 2 more, for a total of 4, to form water. So hydrogen peroxide is looking for 2 more electrons to form water. And fortunately for me and you, we have a way to give the hydrogen peroxide the 2 remaining electrons to form water. And where are they coming from? They're coming from a molecule called glutathione. Now, what is glutathione biochemically? What glutathione is, it's a tripeptide, 3 amino acids in glutathione. It's the one in the middle. That's the important one. The amino acid in the middle of the tripeptide is cysteine. Now, remember earlier in chapter 8, I showed you the structure of cysteine, which has a sulfhydryl group. So now what happens is there is a molecule called glutathione, and you need 2 of them. So I'm going to draw them here. 2G, G stands for glutathione, and the SH refers to the sulfhydryl group of a cysteine in each of the 2 glutathiones. And sulfhydryl groups donate 1 electron. So that's why I need 2 molecules of glutathione. So because each sulfhydryl group of each cysteine of each glutathione will donate 1 electron for a total of 2. And then by doing so, detoxify hydrogen peroxide to form water here. And now when those electrons are given to the free radical, given to the hydrogen peroxide, what happens is the sulfurs become very reactive, and the sulfurs then cross-link with each other, making oxidized glutathione. So reduced glutathione, 2 of them, will donate electrons to the hydrogen peroxide, and then after they're donated, the sulfurs cross-link with each other, forming oxidized glutathione. Now, the name of the enzyme that carries out this reaction, it's a low-yield enzyme, in my opinion, called glutathione peroxidase, which requires selenium. And by the way, this is why selenium is considered as an antioxidant, because it's needed by this enzyme. Now, the fact that it's involved in this antioxidation business, what happens is a lot of students then, or a lot of people, I should say, a lot of people then go to a store and buy selenium pills, thinking, selenium's an antioxidant. I'm going to get rid of my free radicals, so therefore I'm going to go and pop selenium pills. Let me tell you right here at the beginning here, selenium is extremely toxic substance here, a very toxic substance here. There is no reason for anybody to pop selenium pills. Remember, it's a trace element. You don't need very much in the body. There's no reason to pop selenium pills. It is an extremely toxic substance. Now, let me ask you this. In pathology, what is the hallmark of selenium toxicity? Every medical student should know it. How would you know a patient is selenium overdosed? What's the hallmark of selenium toxicity? Anybody know? What's the hallmark of selenium toxicity? That's right. That's right. There it is, Wallace. That's right. It is garlicky odor, the odor of garlic. So, and by the way, arsenic also has a garlicky odor. So selenium and arsenic have this odor. Again, characteristic odor, just like the odor of bitter almonds, diagnostic for cyanide poisoning, garlicky odor, diagnostic for selenium and arsenic poisoning. Later on, musty odor, tomorrow, that's a diagnostic for PKU. So again, the odor is very, very important issues on board exams here. All right, now continue on. Now, if that's all there was, after a short period of time, all of the reduced glutathione would be consumed to oxidize glutathione, and you could not carry out antioxidation again. So in other words, somehow, some way, we got to take oxidized glutathione, give it back its 2 electrons that it gave up, give it back its 2 electrons to restore reduced glutathione to keep the detoxification capacity going. So where are those electrons coming from? Well, they're coming from, there it comes, from NADPH, what comes from the enzyme G6PD, plucking electrons from glucose here. So, and the name of the enzyme that puts the electrons back into oxidized glutathione from NADPH, a low-yield enzyme called glutathione reductase, puts the electrons back into the sulfurs to restore reduced glutathione. Now here it comes. Now to understand what goes on in pathology, in G6PD deficiency, all you got to do is draw arrows up or down. So in G6PD deficiency, the arrow for the enzyme is down. What about the ability to make NADPH? Down. What about the ability to make reduced glutathione? Down. Here, and then what about hydrogen peroxide? Up. So what happens there? Hydrogen peroxide starts to accumulate, and there comes the pathology. They got Heinz bodies and the hemolytic anemia. So now, now you know me, you know we're going to explain this. You know me by now. You know we're going to explain. I'm going to ask you, what is a Heinz body? Here. And what a Heinz body is you've seen these Heinz bodies either in real life or in slides in pathology. But what you're looking at right here, these are erythrocytes. Now, in the erythrocytes, you see these little black spots. And those little black spots are Heinz bodies. And notice that some of these Heinz bodies are all by themselves in the cytoplasm, like you see right here. But many of the Heinz bodies are attached to the membrane, like you see here, there, and there. So, how do you explain that? How does a biochemistry explain that, these Heinz bodies? Very, very, very easy to explain. If you know a little bit about biochemistry, very easy to explain Heinz bodies. And here is the explanation. What's going on there? Suppose suppose this slide in front of you is one single erythrocyte. Now, in the erythrocyte, you have lots of hemoglobin. So here now on the lower left side of the figure, suppose this represents some hemoglobin. They're going to be all over the place, but suppose this represents several hemoglobins. Now on the periphery of the subunits of hemoglobins, yes, there are lysine residues that contribute to the hemoglobin A1C story we talked about earlier. But in addition, on the periphery hemoglobin subunits, you also have cysteine residues. And these SHs are representing cysteine on the periphery of hemoglobin. All normal stuff. This is all normal stuff. You do have cysteine on the periphery of hemoglobin. Here. All right. Now what's going to happen when just think about it. What's going to happen in G6PD deficiency when hydrogen peroxide starts to accumulate? Hydrogen peroxide is an oxidant. Sulfhydryl groups are reductants. What do you think is going to happen? That's right. The hydrogen peroxide can take the electrons from the sulfhydryl groups of hemoglobin to detoxify to form water. Now it's happy. But now the sulfurs of the cysteines, because they gave the electron up, they become very reactive. And what's going to happen is the sulfurs of multiple hemoglobins will now cross-link with each other. And that is now a Heinz body. A Heinz body is those little black spots that you saw. Those are cross-linked, and they denature and they precipitate hemoglobin. So they're cross-linked, denatured, precipitated hemoglobins, multiple denatured by all these disulfide bridges, and that's what those little black spots are here. Now, again, you also saw some of them attached to the membrane. And what happens there is, yes, in the membrane, you also have sulfur lipids. They're called sulfatides. And so sure enough, a hemoglobin can attach to the sulfur lipids in the membrane. Now, what do you think is going to happen in the membrane when you have a piece of junk attached to the membrane? Well, the membrane can rip, and then the cells become very leaky. And as a result, giving rise to hemolytic anemia. Not only that, hydrogen peroxide itself, when it starts to accumulate, can punch holes in the membranes, making the membranes very leaky, hemolytic anemia. Also, the hydrogen peroxide can impair the membrane integrity, and you end up with those bite cells. Again, characteristic of G6PD deficiency. So again, the bite cells, but much more commonly are these Heinz bodies here. Again, major item here. All right, now another item here, and that is where's the? I told you, where's the microbiology at? Where's more pharmacology at? Where is genetics at? As I told you, there's all kind of integration of this disease with all the disciplines. Maybe not behavioral science, but not a science. But where's the genetics at? Integration. Well, since you just had genetics with Dr. Harden a few weeks ago, remember G6PD. The gene for G6PD is located on the X chromosome, and it gives rise to, in mode of inheritance, X-linked recessive, meaning it affects mostly males, G6PD deficiency. But so they can ask about that. They can show you a pedigree of G6PD deficiency. But in addition, the gene for G6PD on the X chromosome displays allelic heterogeneity. Remember, in genetics, what does that term mean? Allelic heterogeneity. That means that gene for G6PD, you can have different kinds of mutations in the same gene. Some people have a mutation of the gene over here. Some people have a mutation of the gene over there. Some people in this world have a mutation of the gene over there. Different kind of missense mutations and so on. That means allelic heterogeneity. So they can ask you about that. And depending on the mutation, they end up with variable expression. They can ask you about that. Variable expression means some people can be severely affected. Some people can be mildly affected. All right, now here comes integration of that term, variable expression, with pathology. Because what that means is, depending on the mutation that some people have with G6PD gene, some people may have 90% of the activity compared to the worldwide population, and they are fine. No problem. Other people in this world may have a mutation in the gene giving rise to 80% of the enzymatic activity compared to the worldwide population. And again, everybody is fine. Then get people in this world who have 70%, 60% of the enzymatic activity compared to the worldwide population. And again, everything is fine until we get to people in this world who have about 50% of the enzymatic activity depending on their mutation of the gene, 50% of the activity, and that's considered in pathology episodic G6PD deficiency, meaning they have just enough enzyme activity to generate just enough NADPH to generate just enough reduced glutathione to detoxify what hydrogen peroxide is normally being made. They're at the maximum capacity to detoxify, and phenotypically they are normal. But if there is some episode that generates too much hydrogen peroxide, for example, infections. Remember in the previous slide, the neutrophils or whatever, if somebody undergoing a bacterial infection, neutrophils are being turned on, generating extra hydrogen peroxide. And here comes the pharmacology. As you know in pharmacology, or should know, there's at least 50 drugs that you should never administer to anybody with G6P deficiency, drugs like sulfa drugs and so on. Why? Because those drugs generate extra hydrogen peroxide, overwhelming the limited capacity to detoxify. So there's certain drugs you never give to somebody with G6P deficiency. But now, in addition, fava beans. Now, people who have episodic G6P deficiency are very susceptible by consuming fava beans. Now, what is a fava bean? Fava bean is like, in case you don't know, it's like a big lima bean. And fava beans are a food staple in the Mediterranean countries. So in Mediterranean countries, they eat a lot of fava beans, but they also have a lot of G6P deficiency, very susceptible to pathology there. Matter of fact, several years ago, when I was teaching on this stuff at my institution, what happened was later on that afternoon, a medical student came up to my office and sat down, and we were chatting. And he said many years ago, when he was a child, he was having a bout of hemolytic anemia. So what happened was, he said his parents rushed him to the doctor, but the doctor could not figure out why, what was the source of the hemolytic anemia. He couldn't figure it out. So then he said they airlifted him to the University of Cincinnati Children's Hospital to figure out what was causing the hemolytic anemia. And they couldn't figure it out there either. And there, he said they were doing blood transfusions on him, keeping him alive and things along that line, until one of the doctors, one of the attendants there, looked at his chart and noticed that his name sounded foreign. It wasn't like Joe Smith. It was a foreign sounding name. So he said, the doctor asked his parents, excuse me, can you tell me what your nationality is? Where are you from? And his parents said, we're from Egypt. Egypt. Mediterranean country. What did you have for dinner last night? And sure enough, indeed, it was a fava bean food. He said his parents almost killed him by giving them a fava bean a meal of some type the night before here. And so now, why am I mentioning this story? Because before you take the USMLEs, it would be to your benefit to get a geography book out and find out what countries are in the Mediterranean countries, so that you know that Ecuador is not a Mediterranean country here. So again, because the way they're going to start the vignette is they'll start the vignette by saying, a 15-year-old Italian male, or a 17-year-old Greek male, X-linked recessive here. And so to give you a clue, again, always pay attention to the first phrase of any vignette. If it's a geographical area, male or female, they give you the age of an individual. If it's a child, usually it's some sort of genetic problem. If it's an elderly individual, usually some sort of nutritional problem, environmental problem, something along that line. But again, the first phrase of a vignette is oftentimes, not always, but oftentimes a clue on what the rest of the vignette is about. So the idea is, if I talk about Italy or Greece or Egypt or Saudi Arabia or Morocco, or you start thinking they could be asking me about G6P deficiency, or it might be about beta thalassemia, something along that line, in which those diseases are somewhat common in that region of the world. High yield. All right, now one last item here. Then we're done with carbohydrates. And that is the issue of where's the microbiology at? Where, I told you the integration with all disciplines. Where's the integration with micro? Now, you just had micro with Dr. Colley just a few days ago, so you should know this. You should know this item here. Now, remember in malaria, in malaria. People who have G6PD deficiency tend to be resistant to malaria. Now, that's a different story from sickle cell. Sickle cell, the beta globin that's defective causes the hemoglobin not to work, and the cells sickle and so on, and they get trapped in capillary beds, and then the sickle cells will eventually lyse. Now, remember in malaria, that a portion of the life cycle of the Plasmodium parasite is the parasite has to go into the erythrocyte to set up shop here. And so what happens is when the merozoite form of Plasmodium parasite comes into the erythrocyte to set up shop, what happens is the parasite then degrades, breaks down all the hemoglobin. The amino acids then are used to make ATP and to grow for the parasite to grow. And eventually the parasite explodes out of the erythrocyte. In fact, that's what causes the fever, when the parasite explodes out of the erythrocyte here. But in sickle cell, again, why people who have sickle cell are resistant to malaria is when the cell sickles, they get trapped in the capillary beds, and the parasite then, when the cell dies, the parasite dies. So again, that's why people who have sickle cell tend to be resistant to malaria. But now is G6PD deficiency, they also tend to be resistant to malaria. Now, why is that? And the answer is from parasitology last week. You should know that all parasites are vulnerable to free radicals. All parasites are vulnerable to free radicals, whether it's malaria, whether it's Plasmodium, whether it's African trypanosomes, whether it's Leishmania, whether it's South American trypanosome, Trypanosoma cruzi, whether it's multicellular parasites like schistosomes. All parasites are vulnerable to free radicals. As an example of this, just to illustrate how important that topic is, those of you from South America, what is the treatment for Chagas disease? And that's acute Chagas, not chronic. There's no treatment for chronic Chagas. But what is the treatment for acute Chagas? Remember, Chagas is where you have Trypanosoma cruzi, infections and so on. What is the treatment in pharmacology? What is the treatment for Chagas disease? Those of you from South America definitely should know this answer. What's in pharmacology? What drug is used? And the answer is it's a drug called nifurtimox. Nifurtimox. There's also a few other related drugs, but nifurtimox is probably the main drug used to treat acute Chagas. So now, why? Why do you give nifurtimox? And the answer is because nifurtimox is a free radical generator. So when you administer this, it generates free radicals, and hopefully it will kill the Trypanosoma cruzi here. In fact, nifurtimox is banned in the United States because it's a free radical generator here. But anyway, that's the idea why you give nifurtimox here. All right, now coming back to malaria. So now, why are they resistant? People have G6PD deficiency. Why are they resistant to malaria? Because when the merozoite form of the parasite comes into the erythrocyte to set up shop, if a person has G6PD deficiency, they have extra hydrogen peroxide around, and the extra hydrogen peroxide will then kill the incoming parasite, and that's why they are resistant to malaria. It all makes sense as long as you know that parasites are vulnerable to free radicals here. It all makes sense. All right. So, all right. So now, all right. Now what's coming up next is how do you treat Chagas in the United States? You don't. You can't treat that in the United States. And by the way, you should know this. In fact, I read a study on this, is that, because of all the immigration problem, here in the United States, that in some of the blood banks, people donating blood, there is some blood that is Chagas positive. So we got to be careful of that, especially in some cities in lower United States, like San Antonio and so on. Some, not all. I mean, some of that blood might be contaminated or be Chagas positive, I should say. So again, got to be really, really careful with Chagas disease. Now in Brazil, they tell me I've been to Rio de Janeiro many, many times, doing a lot of research down there as well as in Sao Paulo and that kind of stuff. But in any case, they say that the Chagas is not a problem anymore, because they do a lot of studies down there. But in any case, just got to be careful of Chagas disease here. All right, so now we're done with carbohydrate metabolism. You see how high yield this stuff is? So now we're done with it. All right, so now we're going to get into now chapter 15. Now in chapter 15 deals with lipids. It says lipid synthesis and storage. Now in this chapter, we're going to be talking about fatty acids to begin with. Then after that, we're going to talk about the rest of the chapter will be on cholesterol. It'll take several hours to cover cholesterol in this chapter here. But first, I want to talk about fatty acids. Now, fatty acids in the human body, you get them from two ways. One way is you can get fatty acids from the diet. But in addition, your body can make fatty acids. Now, in your book, there is a figure that talks about fatty acid synthesis here. Now, fatty acid synthesis is low yield for the boards. So we're going to skip over the figure of fatty acid synthesis. But I want to emphasize there is one major enzyme in fatty acid synthesis, a highly regulated enzyme in fatty acid synthesis that I'm skipping over right now. But trust me, I will cover that enzyme, the top enzyme of fatty acid synthesis, a little bit later in this chapter. And you'll see where I put it in a little bit later in this chapter. But the rest of the pathway of fatty acid synthesis, forget about it. Not important here. There's no pathology associated with fatty acid synthesis. You'd be dead if you can't make fatty acids here. So anyway, so once you get fatty acids in the body, either because you make them or because you get them from the diet, what can the body do with those fatty acids? Well, what the body does with the fatty acids is it makes triglycerides. It puts 3 fatty acids in ester linkage to glycerol, making triglycerides. All right, now right at the beginning here, I want to mention what is an ester bond. You must understand what an ester bond is, because we've got a lot to say about ester linkages. What is an ester bond from organic chemistry? What it is the reaction product of a hydroxy group from one molecule and a carboxy group from another molecule. When they react together, when they condense together, they eliminate water, forming the ester linkage, which you see right here. So that is now an ester bond. Now, if I were to ask you, what's the polarity of a hydroxy group? You're all going to say polar. If I were to ask you, what's the polarity of a carboxy group? You're going to say polar as well. So here comes a question for you. So now the product, the ester linkage, ester bond, what's the polarity of an ester bond? Is it polar or is it non-polar? Let me see your answers here. Is it polar or is it non-polar? That's right. It's non-polar. So in other words, what the body is doing is taking the carboxy group of 3 fatty acids, which are polar. The rest of the fatty acids non-polar, but it takes the polar carboxy groups of the 3 fatty acids and links them to the polar groups of glycerol, and as a result, making 3 ester linkages of 3 fatty acids linked to glycerol. And now, if I were to ask you, what's the polarity of a triglyceride, and the answer is it's 100% grease. 100% grease. There's nothing polar about a triglyceride because it has all these ester linkages. Because in the bigger scheme of life, if I were to ask you, why do you have the adipocytes? And you say, well, the adipocytes is to store triglycerides, which is true. But then why doesn't the adipocyte just store fatty acids? Why do you have to make triglycerides? Why doesn't the adipocyte store fatty acids? And the reason is because that carboxy group would have to be hydrated with water. So therefore, the body makes triglycerides and there's no water, hardly any water in the adipocyte. The adipocyte's less than 10% water because of those ester bonds in triglycerides. Now, the name of the enzymes that make ester linkages in triglycerides are not important. What is important, however, is the name of the enzymes that break down ester linkages of triglycerides. And all those enzymes that cleave ester bonds in triglycerides have a surname called lipases. And there are many, many lipases here. Matter of fact, coming up in this chapter, we're going to be talking about lipoprotein lipase. In the next chapter, we're going to talk about a hormone-sensitive lipase. In GI physiology, you talk about pancreatic lipase. And so by definition, when you see that surname lipase, that's an enzyme that cleaves ester linkages of triglycerides. Now, on the right side here, you also can have ester linkages of fatty acids to glycerol in phospholipids. So here we have two fatty acids and ester linkage to glycerol, and on carbon 3 of the glycerol, you have some head group like inositol, choline, serine, or whatever, making a phospholipid on the right side here. Now, the name of the enzyme that cleaves ester linkages of phospholipids are not called lipases. They're called phospholipases. And there are many phospholipases. For example, there's a phospholipase A1 that cleaves a fatty acid off of carbon 1 of phospholipids. And there's another one called phospholipase A2. That's the one that cleaves a fatty acid off of carbon 2 of a phospholipid. Let me ask you this. Was there any example of a phospholipase A2 that I talked about over the last 4 days? Was there any example of a phospholipase A2 that I covered this week? Yeah, you hear about them in pharmacology. That's true. But was there any example of that that I talked about in biochemistry? Up and down with it, up and down. Where did we talk about a phospholipase A2? Where did we talk about it? Where did we talk about one? What are we talking about? That's right. We talked about it earlier, not in clotting, but in the issue of platelets. Remember platelets, when they bind to exposed collagen, they become activated. They secrete ADP to cause the temporary platelet plug. That's not a clot. That's a temporary platelet plug, but the activated platelets also secrete serotonin. And what does serotonin do? It activates an enzyme called phospholipase A2 that we're talking about right now. And what does phospholipase A2 do? It cleaves off arachidonic acid from phospholipids, and then the arachidonic acid is then made into thromboxane A2, which then does the vasoconstriction. Again, that's not a clot. That's just stopping the blood from flowing at the site of the cut. The clot is generated by the extrinsic-intrinsic coagulation cascades with all that fibrinogen converting to fibrin, and then that we talked about earlier. Now, was there any other phospholipase that I talked about this week, or any other course? Was there any other phospholipase that I covered this week? Oh yeah, up and down with the head. What's another phospholipase that we covered this week? What other one? That's right. That's phospholipase C. Phospholipase C. Now, don't look in the book. But let me ask you this. What receptor is being turned on to ultimately cause phospholipase C to turn on? Like, for example, with epinephrine, what was the receptor for epinephrine that's being turned on? Time out. I told you earlier this week. I know I said this to you earlier this week. When somebody says, or the board say to you, who cares what I say? But if the board says to you, what receptor, blah, 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 don't ever say a G protein. A G protein is never, ever a receptor. Don't ever say a G protein. Here, don't ever say that. I don't know what physiology people are telling us medical students, but G proteins are never, ever a receptor. They're on the wrong side of the membranes, I told you earlier this week. So what's the receptor for epinephrine that ultimately causes phospholipase C to turn on? That's right. It was the alpha-1 adrenergic receptor. Similar receptors for vasopressin, that's true. But the alpha-1 adrenergic receptor for epinephrine is being turned on, and then in turn activates GQ. Alpha Q in turn activates phospholipase C. Remember that? Phospholipase C is being turned on by alpha Q of GQ. All right, now let me ask you this. When phospholipase C is being turned on, that molecule, that the enzyme phospholipase C, then cleaves a lipid called what? What lipid is being cleaved without looking in the book? What lipid is being cleaved by phospholipase C? And that not DAG. No. What lipid is being cleaved? And that lipid is PIP2. And what is PIP2? What is PIP2? You're looking at the answer right in front of you. That molecule right in front of you is PIP2. And so therefore, when you cleave PIP2 with phospholipase C, what were the products by cleaving the bond right here? What's the product of phospholipase C cleavage? One of them is diacylglycerol, shown in the orange color. Di meaning two, acyl means fatty acid. Diacylglycerol, shown there in orange color. And what was the other product? And that in purple, shown in the purple color there, it's the IP3. Again, which then goes to the ER, opens up a channel for calcium, and then calcium, and then DAG, then turn on protein kinase C. Remember all that? Remember all the stuff we talked about? There it is. That's what phospholipase C does. It cleaves that bond right in front of you, generating the two fragments in this signaling mechanism going on with vasopressin, epinephrine, and angiotensin 2. Stuff we talked about earlier. Extremely high yield integration with physiology, endocrinology. Extremely high yield. All right, now what we're going to do is we're going to take a 10-minute break and then come back, and then several hours on cholesterol stuff. Okay, so let's take a 10-minute break and then see you shortly here. Okay, everybody, welcome back for this next hour. So we're on this topic of ester linkages and so on, and how fatty acids, again, are not stored as fatty acids. They're stored as triglycerides in order to, so they don't have to be hydrated. So that's the main reason why you make triglycerides. All right, so now we're going to get into the territory of cholesterol material. And the first thing I want to mention about cholesterol is we got to talk about lipoproteins first. Because you cannot talk about cholesterol unless you talk about lipoproteins. It's like a hand and a glove. They go together here. All right. Now, what is a lipoprotein? What it is, it's a physical complex of lipid and protein. This is in contrast to glycoproteins. Like glycoproteins, it's a covalent linkage of sugar and to proteins here. But lipoproteins are physical complex of lipid and protein. Now, the lipid parts of lipoproteins, they're triglycerides and cholesterol, those type of things, phospholipids. But the protein part of lipoproteins is what I want to cover first here. And the protein part of lipoproteins, they are called apoproteins. Now, in your book on page 223, there is some list of those important apoproteins. But what I want to do here is make it even simpler than what's in your book. I'm going to list the 4 most important apoprotein part of lipoproteins here. Again, there may be about 20, 25 different apoproteins that are known, but only 4 you need to know. The other ones, forget about them. All right, now the 4 apoproteins that you should know, they are apoprotein A, B, E, and C. Now, some of these apoproteins will have Roman numerals after them, like apoprotein A1, A2, A3, apoprotein C2, those type of things. Don't worry about the Roman numeral. It's the letter tells you the function of that particular protein in a lipoprotein here. So again, I'm trying to make it simple. Again, that's the whole, cholesterol is going to get complicated. It's a complicated biochemistry that goes on. So I'll try to make it as simple and understandable as I can, without all the gory details here. But in any case, so these are the 4 apoproteins, important ones. There are other ones as well. Now, what I'm going to do here before I proceed, I'm going to list the function of all 4 of these. Now, as I talk about the function of all 4 of these, if you don't understand what that means, stay tuned, because the next slide is going to be the big picture of lipoprotein metabolism. And you'll see where these apoproteins fit into the big picture once we get to it on the next slide. So if you don't understand what something's going on, don't ask the content TAs yet. Wait till the next slide. That's what we'll talk about, where they're involved and what they're doing here. All right. Now, what apoprotein A is, it's a protein that activates another protein. In fact, it activates an enzyme in the bloodstream called ALCAT. Now, if you don't know what ALCAT does or whatever, don't worry about it yet. But ALCAT is made by the liver only, secreted into the bloodstream, and it does nothing by itself. It has to be turned on by ApoA. Talk more about that on the next slide. Now, ApoB, what ApoB is it's involved in, it's involved in receptor lipoprotein interactions. Now, I want to emphasize there are 2 ApoBs. There's an ApoB48 and ApoB100 on the big picture on the next slide. And those numbers are important. They're not Roman numerals. They are important numbers, ApoB48 and ApoB100, discussed on the next slide. ApoE, there's several of those. Just know them generically as ApoE. They also are involved in receptor lipoprotein interactions. Then finally, ApoC, it's a protein, and what it does, and activates a major enzyme, another major enzyme called lipoprotein lipase, LPL. What that says is LPL, which we touched on a few times already in this course, LPL does nothing unless it's turned on by ApoC. specifically ApoC2. And please keep that in mind because the next couple figures are going to be all straightforward biochemistry, but after that, they're going to be page after page of pathology. And in pathology, you're going to have people who have ApoC deficiency, and other people have lipoprotein lipase deficiency, and they have the same pathology. So either enzyme, either protein not working, then you end up with the same disease. Here. All right. So again, there are other apoproteins as well. All right. Now that's the protein part. Now the lipid part of lipoproteins is you got triglycerides, yes, but in addition, you have cholesterol. All right. Now, cholesterol, there are actually two forms of cholesterol that you can get from the diet. One is cholesterol itself, and the other is something called cholesterol esters. So in your body right now, you have cholesterol in two forms, either as cholesterol or cholesterol esters here. Now, over the years, when I do these reviews, I always ask medical students, what is a cholesterol ester? And so many medical students have no idea what it is. And I tell the students, well, didn't you ever ask your instructor in medical school what a cholesterol ester is? And medical students will say, hey, ours is not to question why. Ours is but to do or die. Well, let me tell you, if you don't know what a cholesterol ester is, we're going to find out because you got to know what a cholesterol ester is. Now, let me ask you this. Before we talk about cholesterol esters, let's talk about cholesterol first. Now, remember earlier, I mentioned to you way back when we were talking about vitamin D, I said to you back then, do you know what cholesterol looks like? Without all the gory details, but in your mind, can you picture what cholesterol looks like? Hopefully, you're picturing 4 rings with all these appendages. And it turns out that cholesterol has 27 carbons. 27 carbons. Now, let me ask you this. Is there anything polar about cholesterol? Let me see your answers here. Is there anything polar about cholesterol? Yeah, no. And the answer is yes. Up and down with the head. Oh, yeah. Something's polar about cholesterol. What is the only polar portion of cholesterol? And the answer is on the 7 o'clock position of cholesterol, specifically on carbon 3, there is a hydroxy group, which is polar. Now, I'm going to show you the structure of cholesterol. The point, again, sometimes it's important to show structures. So I'm going to show you the structure of cholesterol. There it is right there. Has those 4 rings and all these appendages. But at the 7 o'clock position, specifically on carbon 3, there is a hydroxy group. And that's the only polar portion of cholesterol. The rest of the carbons are non-polar. But in carbon 3, that hydroxy group makes it polar. Now, if that's cholesterol, what do you think a cholesterol ester is? We just talked about ester bonds. So what do you think a cholesterol ester is? What's your mind telling you? Well, remember, an ester bond is a reaction product of a carboxyl group and a hydroxy group. Here is the hydroxy group. So something will be linked to it that has a carboxyl group. And what kind of molecules have carboxyl groups? We just talked about fatty acids. And sure enough, so when you have a fatty acid in ester linkage to cholesterol, that's now called a cholesterol ester. And that fatty acid shown there in red can be any fatty acid. It could be arachidonic acid, it could be stearic acid, palmitic acid, oleic acid, linoleic acid. It could be any fatty acid. In ester linkage to cholesterol is generically called a cholesterol ester. All right, now if I were to ask you, what can you tell me about the polarity of a cholesterol ester? Is there anything polar about a cholesterol ester? Anything polar about it? That's right. It's 100% grease. Just like a triglyceride, a cholesterol ester would float on water. And so because there's nothing polar about it. And by the way, that's going to be a big time problem in humans because when you have cholesterol esters in the aqueous environment called your bloodstream, they don't like each other. Water and cholesterol esters, they don't like each other. And that's going to be a serious problem in human beings here. All right. Now, so when you eat cholesterol in those 2 forms, cholesterol and cholesterol esters here. All right. Now, one more thing before we get to the big picture of lipoprotein metabolism. Let me mention that it's going to get hairy, if you never understood cholesterol and lipoprotein metabolism, again, I'll take it nice and slow. No need for me to rush through this. I'm going to take it nice and slow, but please be motivated to want to know this material because it's not going to be easy, but it definitely will be understandable if you're motivated to understand the material here. But anyway, give me a chance to explain what goes on here. All right, now in your book around page 224 and so on, there's several figures that talk about cholesterol metabolism. I'm all going to boil it down to this one slide on page 224. Now, that one figure there. Now, what you're looking at is the top of the figure, and after we go through a thorough discussion of that, then we'll do the bottom of the figure on page 224. But right now, you're seeing the top half of the figure because I'm going to focus your attention to what happens to dietary lipid. So that is not the top of the figure you see here in front of you. That is what happens to dietary lipid here. All right. Now on the upper left-hand side of the figure, here is the intestine. Now remember from GI Physiology from Dr. Wilson's review, or some of you will get Dr. Wilson a few weeks from now, depending on which block you took. But in any case, the idea there is when you eat a high-fat meal, again, nothing happens in the mouth or stomach until the food enters the intestine. And then remember the intestine, you have enzymes called pancreatic lipase, co-lipase that break down triglycerides, the fatty acids. And you also have enzymes called pancreatic esterases that cleave the fatty acid off cholesterol here. Because remember, because triglycerides cannot go through a membrane. They're too greasy. You might imagine cholesterol esters cannot go through a membrane either because they're too greasy here. And as a result, you have enzymes, pancreatic enzymes that break down triglycerides, break down cholesterol esters to fatty acids plus cholesterol. And then what happens is then you absorb the fatty acids, you absorb the cholesterol into the mucosal cell. What happens next? Let me see your answers quick. Let me see your answer here. In the mucosal cell from GI Physiology, after you absorb the fatty acids, after you absorb the cholesterol, what happens to them? Let me see your answers here. No, not packed in chylomicrons yet. No. What happens to fatty acids and cholesterol? You reform them into triglycerides and cholesterol esters. Here. So again, you break down triglycerides in the intestinal lumen, you break down cholesterol esters, intestinal lumen, and then by doing so, you absorb the fatty acids, absorb the cholesterol into the mucosal cell. After the absorption, then you reform the triglycerides and reform the cholesterol esters. Before you do anything else, you reform them. Here. Again, it seems like a waste. You're breaking down triglycerides and cholesterol esters, but they cannot go through the membrane here. And all right, now, then after you reform the triglycerides and after you reform the cholesterol esters, then you package all that grease into a massive particle called the chylomicron here. And the chylomicrons, again, now, by the way, just to put things into perspective, the amount, according to nutritional experts, the maximum amount of triglycerides you and I should eat every day, the maximum amount, according to them, should be 80 grams of triglycerides. 80 grams of triglycerides here. And by the way, if you eat one hamburger, like a Big Mac is about 34 grams of fat. If you go to Burger King, a Whopper without the cheese is about 39 grams of fat. So if you eat one hamburger, roughly you're eating half of what you should eat a day, according to nutritional experts. 80 grams. Now, in terms of cholesterol and cholesterol esters, according to nutritional experts, the maximum you and I should eat is 300 milligrams. It's about 1,000 times less than triglycerides. Gram amounts of triglycerides, milligram amounts of cholesterol and cholesterol esters here that you and I should eat maximally per day. And to put that number into perspective, 300 milligrams of cholesterol, If you eat one egg, in one egg yolk, there is 250 milligrams. So if you eat one egg, basically you're at your maximum. If you eat a 3-egg omelet, you're way over what you should be eating per day. So again, so gram amounts of triglycerides, milligram amounts of cholesterol and cholesterol esters here. And you package all that grease in a massive particle called the chylomicron. Now, a chylomicron is about 98% lipid. It's a huge particle. And of all that lipid, it's gram amounts of triglycerides and milligram amounts of cholesterol esters here. No cholesterol, it's one to mucosal because you have cholesterol in the mucosal cell. And on the surface is a protein called ApoB48. Now, remember from GI physiology, these chylomicrons are first sent into the lymphatics. And then from the lymph, then eventually the chylomicrons will enter the bloodstream. Now, before we get to that issue, like entering the bloodstream, what I want to do is now integrate this with pathology. In pathology, what is the name of that genetic disease in which there's a gene defect of ApoB48 such that the chylomicron cannot be assembled in the mucosal cell? So again, there's a gene defect in B48. What's the name of that all-important disease? What's the name of that disease? Not hypercholesterolemia. We're not talking about the bloodstream yet. We're not talking about the blood. Hypercholesterolemia means the bloodstream. This is before the bloodstream. What's the name of that disease? And that is called A-beta-lipoproteinemia. Now, what's going on with the bloodstream here? So this disease, A-beta-lipoproteinemia, again, it's due to a defect in ApoB48. As a result, it's characterized by having abnormally low levels of serum triglycerides and serum cholesterol. So there is A-beta-lipoproteinemia because low levels of serum triglycerides and abnormally low levels of serum cholesterol. Not hypercholesterolemia. That's a different story. Here, too much cholesterol in the bloodstream. Here, it's just the reverse. Low levels of cholesterol and triglycerides in the blood. Here. All right, now, let me ask you about A-beta-lipoproteinemia. Now, this disease, it's characterized by having, by the way, let me ask you this. So, in this disease, if a person eats a high-fat meal or whatever, then they break down the triglycerides and so on, cholesterol, and they get absorbed into the mucosal cell. But now the mucosal cell cannot assemble the chylomicron because ApoB48 is defective. What happens to all that grease? What happens to all that grease in the mucosal cell? If it's not packaging chylomicron, what happens to it? What is this disease characterized by? What's it characterized by? That's right. It's characterized by severe steatorrhea. Steatorrhea. Why? Because that grease has to go somewhere, and all those mucosal cells will slough off, and all that grease that's in them will eventually go into the stools, and then the stools, again, characterized by severe steatorrhea. Remember, the stools maximally should be 6% grease here. But if it's over 6%, then the stools look greasy here. In fact, on board exams, they talk about floating of stools of a child or whatever, floating stools in a toilet, something along that line, saying, well, that's a lot of grease. They're floating, and then that's probably indicative of steatorrhea. So this disease is characterized by severe steatorrhea. All right, here comes the question for you. Besides that pathology of steatorrhea, again, they have low levels of serum triglycerides and cholesterol. Other than the steatorrhea, is this disease relatively benign, or is this a very severe disease characterized by low levels of serum triglycerides and cholesterol? Let me see your answers here. That's right. It is very, very severe disease. Now, why is that? Why is that is if I were to tell you right now that these people with A-beta-lipoproteinemia, they have night blindness, They have bleeding problems. They have severe developmental issues that go on. So how would you explain all that severe pathology? You explain that by saying that in the chylomicron, besides triglycerides and cholesterol, that you also have the 4 fat-soluble vitamins. And therefore, if you do not assemble the chylomicron, the 4 fat-soluble vitamins will not be delivered. Therefore, vitamin A, no vitamin A, night blindness, no vitamin K, bleeding problems, and so on. So again, because of the 4 fat-soluble vitamins. But now, a much harder answer is this. Much harder answer. How would you explain the severe developmental issues in a beta-lipoproteinemia? Does anybody know there? How would you explain that? Kind of a hard answer. How would you explain the severe developmental issues? Hormones, fat-soluble vitamins. We just talked about the fat-soluble. How would you explain it? And here comes the answer. Remember, even though I skipped over at the beginning of the chapter, I'm going to cover it right now because I knew that's why I skipped it over. I'm going to cover it right now. Remember, there are 6 essential lipids the body cannot make, has to get them from a diet. 4 of the 6 are the 4 fat-soluble vitamins. What are the other 2 lipids that are essential? The body must get them from the diet because the body cannot make them. What are the 2 other essential lipids? And that's right. They are the 2 essential fatty acids, the 2 L's, linoleic and linolenic acid. Again, that's discussed at the beginning of the chapter, but I'm talking about it right now. So you need 6 essential lipids that are in this chylomicron. That have to be delivered to the other cells in the body. The 4 fat-soluble vitamins and the 2 essential fatty acids, linoleic and linolenic acid here. All right. So what? What's that got to do with the severe developmental issues? Who wants to connect the dots for us? Who wants to tell me why, if there's no linolenic acid, they end up with severe developmental issues? How do you explain that? You explain that by saying because linolenic acid are needed to make arachidonic acid, and arachidonic acid, which is not essential, because you can make it from linolenic acid, and so you make arachidonic acid from them, and arachidonic acid is needed to make prostaglandins, and therefore no linolenic acid, you will not make arachidonic acid, you will not make prostaglandins. Severe developmental issues that go on. It makes sense of what goes on there. Again, that is what's going on there. Again, all the integration with pharmacology, with the prostaglandins and what they do, they come from arachidonic acid, which in turn comes from the essential fatty acids here. Okay, all right, now continuing on, assuming everything's fine, assuming normal biochemistry now. So once the chylomicron is assembled, a massive particle in the mucosal cell, is sent into the lymph, and then via the thoracic duct, the chylomicrons then enter the bloodstream. And when the chylomicrons enter the bloodstream, they acquire ApoC and ApoE on their surface here. ApoC and ApoE is on the surface. Let me ask this question. Where did they come from? Where did ApoC and E come from? Where did they come from? That's right. They come from donation from HDL. In other words, when a chylomicron enters the bloodstream, as blood circulates, chylomicrons and HDL are already in the blood. What happens is as blood circulates, they're banging into each other, colliding with each other as blood circulates. In a momentary period of time, when a chylomicron bangs into an HDL, HDL is going to give gifts of ApoC and ApoE to the circulating chylomicron. That's how the chylomicron gets its ApoC and E. And then HDL goes away. Now, what happens to the chylomicron in the bloodstream? Well, what happens is when it gets into the vicinity of the adipocyte, on the surface of the endothelial cells that line the adipocyte, there is that major enzyme again, lipoprotein lipase. And what's this enzyme going to do? The name of the enzyme tells you what it does. Because it's a lipase, it's going to cleave triglycerides from the chylomicron, and then the fatty acids are absorbed. The fatty acids are absorbed into the adipocyte. They're put onto glycerol and then to reform the triglycerides. And then the triglycerides are then stored in vesicles, waiting for the rainy day to occur here. All right. So again, the triglycerides break down because they cannot go through the membrane. That's why you need ApoC to turn on lipoprotein lipase. You break down triglycerides and fatty acids. The fatty acids put back on the glycerol, and then reform the triglycerides. They're now being stored, waiting for the rainy day. Now, let me ask you this: in endocrinology, from Dr. Wilson's reviews, or will review for you, in turn, the adipocyte, when all these triglycerides reform in the adipocyte in turn sends out another hormone. What hormone is being sent out by the adipocyte? What hormone is being sent out by the adipocyte? Anybody know what hormone is being sent out by? That's right. Not lectin, it's leptin. Leptin is being sent out by the adipocyte, leptin. And what does leptin do? Then leptin, what's its target cell or organ or whatever? What is the target of leptin? That's right. And the answer is the target of leptin is the hypothalamus. So leptin binds to receptors on the hypothalamus, and then neuropeptides are then released in order to cause the cessation of eating here. So that's a feedback situation that goes on. The leptin binds to receptors on the hypothalamus, which in turn signals the release of neuropeptides, which then causes the body to not want to eat very much. It goes on there. All right. So again, that's the feedback there. Now, coming back to the triglycerides that are now being stored in the adipocyte, coming from the diet here. So the triglycerides are now being stored, waiting for the rainy day. Now, here comes a question for you. When does the rainy day begin? When does the rainy day begin? When does the rainy day begin? Don't say starvation. I know that. Everybody knows starvation. I'm talking about time. I want time. When does the rainy day begin? You're hurting me. You're hurting me. You're hurting me. Because we talked about this earlier. You're hurting me. When does the rainy day begin? 90 minutes after the last bite of carbohydrate in the meal. Remember that all-important glucose tolerance curve we talked about and I hammered home the other day? I said you better know this graph. 90 minutes after the last bite of carbohydrate in the meal. Then the body switches to page 163. And on page 163, you start to send out hormones because you got to maintain blood glucose. You got to turn on gluconeogenesis and you got to turn on, and gluconeogenesis requires energy and coming from fatty acids. And sure enough, the rainy day begins 90 minutes after the last bite of carbohydrate in your meal. Not any meal, carbohydrate in the meal, because that's when the blood glucose drops below 5 millimolar. The body switches to page 163. I spend a lot of time talking about, and you're hurting me because I drove home that point. That's when the rainy day begins. Please keep that in mind, because that's when the body switches to page 163 and all the stuff about endocrinology and mobilizing this and mobilizing that. We covered that already, and you're hurting me because I drove home that point. Any case, let's continue here. Now, what happens to the chylomicron when all these triglycerides are being removed? Well, the chylomicron starts to shrink and shrink and shrink because triglycerides are being removed. And somewhere during the shrinking process, then when it encounters HDL again, then the chylomicron will then give to the HDL its ApoC. So the ApoC was used, but not abused, and now given back to the HDL. And at that point there, the chylomicron then converts to what's known as the chylomicron remnant. Now, what's the chylomicron remnant? It's much, much, much smaller in size compared to a chylomicron because most of the triglycerides were removed. So the chylomicron remnant just has residual, not much, just has residual triglycerides remaining. It still has the cholesterol esters in it. Here, nothing happened to the cholesterol esters. And on the surface is ApoB48 and ApoE because most of the triglycerides were removed. So again, the chylomicron is gram amounts of triglycerides and milligram amounts of cholesterol esters. But now the LPL enzyme removes most of the triglycerides, so ending up as a chylomicron remnant. As the chylomicron shrinks and shrinks and shrinks all the way down to the chylomicron remnant, which at that stage just has residual triglycerides remaining and still has the cholesterol esters. Nothing happened to them, and then ApoE and B48 on the surface. Now, what happens to the chylomicron remnant? What happens is it's removed out of circulation by the liver and liver only because the liver has a receptor called the remnant receptor. Now, again, what's the function of ApoB and E? To be involved in receptor lipoprotein interactions. So docking of the remnant with a remnant receptor occurs. Now bring in the cell biology histology. Remember, when docking occurs, then what happens is endocytosis begins, and then the endosome then fuses with lysosomes, and then the lysosomal enzymes then break down whatever came into the endosome. So sure enough, proteases break down the ApoE and ApoB100 down to amino acids. But in addition, in lysosomes, there is an enzyme called a lysosomal esterase, that'll cleave off the fatty acid from cholesterol esters. So again, the chylomicron remnant is now bringing into the endosome cholesterol esters, but the lysosomal enzymes will now cleave off the fatty acid from the cholesterol esters. And as a result, now we have incoming cholesterol in the liver here. It came in as a cholesterol ester. You hack the fatty acid off in lysosomes, and now you have incoming cholesterol in liver cells. All right, now let me just summarize thus far. So what this says is, for all intensive purposes, all dietary triglycerides end up in the adipocyte. All dietary cholesterol ends up in the liver. That's what this says. And you see how it happened. You see the biochemistry, how it all happened. All dietary triglycerides end up in the adipocyte. All dietary cholesterol ends up in the liver. So then the question is, okay, well then what's the liver going to do with all that grease, all that cholesterol it got from the diet, milligram amounts of it? All right, now to understand what the liver is going to do with the cholesterol it got from the diet, now let's bring in the rest of the story. All right, bring in the rest of the story here. So now the rest of the story is what happens to the body lipid. So the first part was dietary lipid, but now the rest of this figure is body lipid. What does a body do with body lipid here? All right, let's focus in on the liver. All right, now the liver again is getting milligram amounts of cholesterol from the diet, but as we'll talk about at the end of the chapter, the liver can also make milligram amounts of cholesterol in a pathway called cholesterol synthesis. So what's the liver going to do with all that cholesterol? It got from the diet or it makes. But now, in addition, the liver, if you eat a high carbohydrate meal, the liver can take that glucose, all the excess glucose will come into the liver by a first order transporter, GLUT2. And then once glucose enters the liver, then the glucose will then convert all the way down to pyruvate, down to acetyl-CoA by biochemistry that we talked about earlier. And then in the liver, the acetyl-CoA will then see a major enzyme in fatty acid synthesis, a mega enzyme there called acetyl-CoA carboxylase. Remember, I told you we're skipping over fatty acid synthesis figure, but there's one important enzyme, and there it is. That enzyme is the acetyl-CoA carboxylase. This is the enzyme, as the name says. What it does is it takes the acetyl-CoA coming from the glucose that you eat. It takes the acetyl-CoA and carboxylates it. It puts a CO2 onto it to make a 3-carbon product called malonyl-CoA. So malonyl-CoA is 3 carbons, and then multiple malonyl-CoAs then go on to make fatty acids in fatty acid synthesis. Don't worry about the details. I'm not going to ask you about how that happens. That's low yield. So again, one more time. So when you eat a high carb meal, then the glucose then in glycolysis goes down to pyruvate, down to acetyl-CoA. And then this enzyme kicks in, acetyl-CoA carboxylase, which uses biotin, remember carboxylase enzymes uses biotin as a coenzyme to convert acetyl-CoA 2 carbons to the 3-carbon malonyl-CoA. And then multiple malonyl-CoAs then start to condense together to form a fatty acid in the liver here. Now that enzyme, acetyl-CoA carboxylase, why is it so important? And the reason is it's hormonally regulated. What hormone makes sense to you turns on the enzyme? You might imagine it's going to be insulin. Insulin turns it on at the gene level. Also, insulin turns it on by dephosphorylation mechanisms. Any enzyme that might be around, acetyl-CoA carboxylase enzyme that's around, insulin results in its dephosphorylation, turning the enzyme on. All right, now remember, in the liver, insulin has major effects. Remember, in the liver, insulin has nothing to do with glucose uptake. Nothing to do, because it's a different transporter, GLUT2. But once insulin takes command, once glucose enters the liver, insulin takes command. It drives glycolysis and down to acetyl-CoA. Then the acetyl-CoA in turn, insulin turns on the top enzyme there to drive fatty acid synthesis. This is why most fatty acids in the body are made by the liver. Yes, other cells, even the brain can make a little bit of fatty acids, but 99% of the fatty acids are made by the liver. Why? Insulin mediation. So again, in the bigger picture. All right, so now what is the liver going to do with a milligram amounts of cholesterol it got from a diet or it makes, and the gram amounts of cholesterol, triglycerides that the liver made from the glucose? So what is the liver going to do with the milligram amounts of cholesterol and the gram amounts of triglycerides? What is the liver going to do with all that grease? Well, the strategy, the game plan is the liver wants to take all this grease and then package it and send it out in circulation, and then send all the triglycerides to the adipocytes for storage that it made. And then the other strategy, the other game plan is the liver wants to take the cholesterol that it got from the diet or it makes and ship it off to the other cells so the other cells don't have to make the cholesterol. Because as we'll talk about at the end of the chapter, cholesterol synthesis is expensive. It takes a lot of ATP and carbon to make cholesterol. As I told you earlier, the liver is a wonderful and gentle organ. The liver is saying to the community of organs out there, hey, adrenal cells, for example. Hey, adrenal cells, I know you need cholesterol to make some steroid hormones, but don't make the cholesterol. I got your back. I'm going to send you cholesterol that I got from a diet or I made. I'm going to send you, the adrenal cells, some cholesterol. So don't make it. Don't waste your effort to make it. I got your back. I'm going to send it to you. So that's the idea. The bigger picture here. The liver is sending the triglycerides to the adipocytes for storage, and the liver is sending the milligram amounts of cholesterol to the other cells, hoping they don't have to make it. They can use it to make steroid hormones or other things. So that's the idea. That's the strategy here. But as we'll find out in a little bit, the other cells may not want that cholesterol. And if the other cells don't want that cholesterol, big time problem. Something somebody mentioned earlier called the hypercholesterolemia, because the other cells may not want that cholesterol. Big time problem here. All right, so that is the strategy here that goes on in the liver. Now, before we talk, so now the liver is going to package all this grease, all the cholesterol in milligram amounts, and triglycerides in gram amounts, and package it with a protein in another big particle called the VLDL, which stands for very low density lipoprotein. Here. Now, VLDL, it's a pretty big particle, not quite as big as a chylomicron, but nevertheless, it's a big particle. It's 90% lipid. Chylomicrons are 98% lipid, but VLDL is 90% lipid. And of that lipid in it, it's mostly triglycerides that the liver made. By far, that's the dominant molecule in the VLDL are triglycerides. So it's gram amounts of triglycerides in VLDL and milligram amounts of cholesterol. And notice the cholesterol that's in VLDL. It's cholesterol. Not a cholesterol ester yet. When cholesterol leaves the liver, there is no fatty acid on it yet. Now, if you think that's trivia, you better be here in the next hour because we've got a lot to say about that here. So when cholesterol leaves the liver, there is no fatty acid on it yet. That is a big issue coming up soon here. When cholesterol left the intestine in chylomicrons, there is a fatty acid on. But when it leaves the liver, there is no fatty acid on it yet. Now, the protein that's on the VLDL is a protein called ApoB100 on the surface. It's related to ApoB48 in the intestine, but this is B100, ApoB100 here. It's on the surface of VLDL. Now the VLDL is sent directly into the bloodstream, not in lymphatics. It's sent directly into the bloodstream here. Now, before we proceed and discuss what happens to the VLDL with all of its grease, what happens to it? Before we talk about it, let's integrate with pathology, this figure with pathology. Now, everybody knows, if I were to ask you right now, what does the liver look like in an alcoholic? Everybody knows this, should know this answer. What does the liver look like in a long-term alcoholic? That's right. It looks fatty. It looks fatty. So now the question is why? Why is the liver fatty in an alcoholic? Now, what happens is a lot of medical students will say, well, I know why. It's because alcohol is highly caloric. It has 7 kilocalories per mole. It's highly caloric and making a lot of fatty acids and triglycerides, which is true. That's all true. There's no question that is true. Ethanol is highly caloric, but that's not causing the fatty liver. Because after all, if the liver takes all that ethanol and makes fatty acids and triglycerides, just ship them off to adipocyte for storage. But now what's causing the fatty liver, however, is this. Yes, the liver is going to take ethanol and makes a lot of fatty acids and triglycerides. But the problem there is alcohol inhibits VLDL assembly. Alcohol is dissolving the VLDL, and therefore it cannot assemble the VLDL. And as a result, all those triglycerides cannot exit the liver. They cannot go through a membrane. They cannot exit the liver, and all these fatty acids will now back up into the liver, forming in vesicles, and liver then looks fatty here. Now, what you're looking at right here on the left side is a normal rustic colored liver. On the right side is a liver in an alcoholic, much lighter color. And if you do a histological examination, these are normal hepatocytes, and on the right side. And these are hepatocytes with all those lipid droplets, the triglyceride droplets in the interior of the hepatocytes, and all those. And so they're sitting in these vesicles with all those triglycerides here. Now, how long does it take for the liver to look like this with all these lipid droplets? It takes something like 2, maybe 3 weeks of binge drinking. 2, maybe 3 weeks of binge drinking. Your liver will look like this. Now, fortunately, it's reversible at this point. But if a person continues to drink alcohol, as you might imagine, eventually the liver becomes scarred, and then ultimately get into the very serious alcoholic cirrhosis that goes on. Now, let me ask you this. In histology, what is the name of the cell type in the liver that's scars the liver and giving rise to their irreversible alcoholic cirrhosis. What's the name of this cell type? That's right, Annie and Juliana, that's right. It is the Ito cell, the ITO. The Ito cell is the name of the cell type that scars the liver. And when that happens, it is a very, very serious problem that goes on there. Okay, so anyway, so again, I did want to cover some pathology here with alcoholic cirrhosis that goes on here. All right, now assuming everything is normal, again, no alcohol business here, but suppose everything is normal, then the liver then packages all this grease in this massive particle called the VLDL. All right, now what happens to the VLDL? Well, what happens to the VLDL in the bloodstream? It'll encounter HDL. Then HDL, when it bangs into HDL, HDL will give gifts to the VLDL. HDL is like Santa Claus. HDL is giving gifts of ApoC and ApoE to the circulating VLDL. Then HDL goes away. Now what happens to the VLDL? Because now on its surface, it's decorated with ApoB, ApoC, and ApoE. So what happens to the VLDL? As it circulates, it'll encounter, the ApoC will then encounter lipoprotein lipase again. So ApoC of VLDL will turn on LPL. Triglycerides break down from the VLDL that the liver made, and then fatty acids are absorbed into the adipocyte, put onto glycerol, and then stored in vesicles in the adipocytes here, waiting for the rainy day. So again, triglycerides are broken down by the all-important enzyme lipoprotein lipase here. All right, now the question is this, here, is now LPL also, you need to know, besides being turned on by ApoC, also is hormonally regulated. What hormone turns on lipoprotein lipase? We talked about it earlier, but what's the name of the hormone? That's right. That again is insulin. Insulin again is now turned on LPL. All right. Now, before we take the 10-minute break, let me summarize what insulin is doing. Because over the last couple of days, we talked about bits and pieces of what insulin's doing here, insulin's doing there, and this kind of stuff. Let me put it all together in the bigger picture of life, what insulin is doing here. Here it comes. And again, you should be able to do this. You should be able to verbalize. If I ask you right now, what is insulin doing here? And you should be able to verbalize, and here it comes. The idea is this. When you eat a high carbohydrate meal, all the excess glucose will funnel into the liver. Yes, glucose enters all cells, but all the excess glucose pours into the liver by a first order transporter called GLUT2. And insulin has no effect on that. But once glucose enters the liver, insulin takes command of the liver, and the first thing insulin does to the liver, it tells the liver, hey, liver, turn on the gene for glucokinase to trap the incoming glucose. And sure enough, that glucose will now be trapped as glucose 6-phosphate, all in the direction of insulin mediation. And then once glucose 6-phosphate is generated, insulin does another thing in the liver. Insulin then will tell an enzyme called PFK2 to make a product to override the inhibition of PFK1. And so by override inhibition, then glycolysis now works. How insulin is now directing glycolysis to work overtime in the liver. Whereas in other cells, in a high carb meal, glycolysis is coming to a grinding halt, but in the liver, it's working overtime, driving glycolysis all the way down to pyruvate, then down to acetyl-CoA. And then insulin does now another thing in the liver. Insulin now will tell another major enzyme called acetyl-CoA carboxylase to turn on. And when it turns on that enzyme, you're going to make a fatty acid precursor called malonyl-CoA. Then multiple malonyl-CoAs will then condense to form fatty acids in the liver, all under the direction of insulin going on in the liver. While all that's going on in the liver, meanwhile, back at the ranch in the adipocyte, Insulin will come to adipocyte and tells adipocyte, hey, adipocyte, get ready with your lipoprotein lipase at the gene level. Make the enzyme and put it on the surfaces, and because soon triglycerides are coming your way in the form of VLDL. Hey, adipocyte, get ready with GLUT4. Put it onto the surface because we need to bring in more glucose into the adipocyte to metabolize that glucose down to glycerol to get the glycerol platform to put the fatty acid on. How insulin is directing the show. How insulin's telling these organs what to do in the liver and in the adipocyte when you eat and when a person eats a high carb meal. And you can only appreciate diabetes, the pathology of diabetes, when you don't have the insulin effect. The liver doesn't know what to do with all that excess glucose. The adipocyte does not know what to do either. And it is a mess. That's why diabetes is such a messy disease when you don't have the insulin effect. Then all that glucose stays in the blood, and we talk about what happens to it in the blood, causing havoc to the vasculature. Let me just think about the biochemistry here. One little polypeptide hormone, a little bitty hormone, is how you gotta have it, and it gotta work biochemically properly in order for these organs to work. And if you don't have insulin around, or if it's a receptor not around, whatever, it is a big time problem in the body here. All right, now what we're going to do is we're going to take another 10-minute break and then come back after the 10 minutes and talk about more biochemistry on this slide, next slide, and talk about what happens to VLDL when triglycerides are being removed. Okay, 10-minute break, and then see you shortly here. Okay, everybody, welcome back. It said some of you said my mic wasn't muted. It looked like it was muted. I mean, the red button was on and all that, so I'm not sure why that says it wasn't muted. But in any case, hopefully you can hear me right now. Oh, you muted it. Okay. Okay, thanks, Maggie. All right, Maggie muted it for me. Here. All right. So anyway, we're on this topic. We're on this topic of what happens to the VLDL. So again, the VLDL is bringing triglycerides to the dipeptide site for storage, and then ApoC turns on lipoprotein lipase. Triglycerides then break down, and then the VLDL is shrinking because triglycerides are being removed. Now, during the shrinking process, the VLDL will encounter HDL again, and then it'll give back this its ApoC. So again, ApoC was used but not abused, and then given back to the circulating HDL. And at that point there, then that huge VLDL particle shrinking, then converts to what's now known as an IDL particle. And what's IDL? It stands for intermediate density lipoprotein. It just has residual triglycerides remaining, still has the cholesterol, no fatty acid on it yet. It has ApoB 100 on the surface, ApoE on the surface as well. So again, IDL, it's a much smaller particle than VLDL. It's a shrinking all the way down to the IDL particle here. Okay, because it gave up its ApoC back to the circulating HDL. All right, now what's the fate of the IDL particle? What can happen to it? Well, what happens is two things can happen to the IDL particle. One fate of IDL is to be removed out of circulation by the liver only. The liver has yet another receptor. Don't worry about the name of this one, but it's another receptor to which IDL will dock with. Then IDL will be removed out of circulation by that receptor in the liver only. Now, the other fate of IDL is coming up right now. This is the one we really need to pay attention on and think about it carefully, because this is the part that could be a little confusing. What happens to the other fate of IDL? When it encounters HDL again, when it bangs into HDL again, IDL will give to the HDL its cholesterol. So again, the cholesterol is now being transferred from IDL onto HDL. And what's HDL going to do with it? Well, HDL is going to be involved in the process of taking the cholesterol, putting the fatty acid onto it to make a cholesterol ester. Once it makes a cholesterol ester, then when HDL encounters IDL again as blood circulates, then HDL will give to the IDL cholesterol esters back to the circulating IDL. Incredible biochemistry. This is as blood circulates. That cholesterol is like a hot potato. These lipoproteins are saying, you take it. No, you take it. No, you take it. It's going back and forth from these lipoproteins. Incredible biochemistry as blood circulates. Now, the obvious question is, repeat again, as when the VLDL is shrinking down to the IDL, it's given ApoC back to the HDL. But in addition, as blood circulates, IDL can give to the HDL its cholesterol. Cholesterol will now be involved in the process of putting the fatty acid onto the cholesterol to make a cholesterol ester. And then after it makes the cholesterol ester, when HDL encounters IDL again, HDL will give to the IDL cholesterol esters back to the IDL particle here as blood circulates. The obvious question is, why are you doing this? And to explain the why, very easy to explain. Why are you putting the fatty acid on the cholesterol to make a cholesterol ester? Now, after we do the why, then we'll do the when, where, and how, why you transferring the HDL, all that kind of stuff like that. But the first things first, Why are you putting the fatty acid on the cholesterol? And again, easy to explain. Here is the reason. Here is the answer. Suppose on the left side here, suppose that represents any lipoprotein, a generic lipoprotein. Now remember, on the surface of lipoproteins, there is no membrane. It's simply anything polar, like proteins, will be on the surface of a lipoprotein, facing the water environment called the bloodstream. Anything greasy, like triglycerides, will be in the interior. It's not that triglycerides like each other. It's simply triglycerides don't like water. So they'll be found in the interior of the lipoprotein. All right, and here comes a question for you. Now, in a generic lipoprotein, where would cholesterol be located? On the surface or in the interior? There's no membrane. Kevin, there's no membrane here. There's no membrane in lipoproteins. What would cholesterol be found? On the surface or in the interior? Where would cholesterol be located? On the surface or in the interior? And the answer is it will be found on the surface. Why? Because of that single hydroxy group here. Because it's polar, it faces the water environment. So in other words, suppose on the lower left here, suppose this represents two cholesterol molecules. There are going to be many more on the surface, but this represents two cholesterol molecules. And each of them has a hydroxy group that faces the water environment. The oval part represents cholesterol, facing the interior because it's greasy here. So cholesterol is on the surfaces of your lipoproteins. Now remember from first year biochemistry in your medical school, hopefully your instructor told you that in the plasma membranes of all the cells in the body, besides phospholipids, you also have cholesterol in your membranes. All your membranes in your body, you have cholesterol in your membranes here. So here we have cholesterol. So what you see here in the blue, suppose that PM stands for plasma membrane of any cell, and in the membrane, you also have cholesterol. Now, why do you have cholesterol in your membranes? You know, that's the thing about cholesterol, which is unfortunate for cholesterol. It doesn't get the respect it deserves. You know, if you ask your relatives, you ask your friends, what do you think about cholesterol? Most people say, ooh, cholesterol, bad news, bad news. I'm telling you right now, cholesterol is essential for life. You gotta have cholesterol in your body. Why? For two reasons. Why is cholesterol essential for life? One reason is it's the precursor of all steroid hormones. And secondly, it's the key modulator of membrane fluidity. In other words, in your membranes, you have all these phospholipids, but the fluidity of the membrane is tightly regulated. It cannot be too fluid nor too rigid. Why? Because in your membranes, you have all these receptors, you have all these transporters that are always moving things in and out of cells and things along that line. And these receptors have to move laterally across the membranes and things like that. And the membrane has to be properly fluid, not too rigid and not too fluid. And what regulates membrane fluidity? It's cholesterol. Cholesterol is the key modulator of membrane fluidity. And the amount of cholesterol in the membranes is tightly regulated. Not too much, not too little, to regulate the membrane itself here. All right, now that's what they should have told you in first year biochemistry in your medical school about cholesterol in your membranes. All right, now look at the lower left here. So now as blood circulates, when lipoproteins are circulating in somebody's bloodstream, they're always banging into cells. Lipoproteins are banging into cells. Now, in a momentary period of time, when a lipoprotein bangs into a plasma membrane of a cell, what can happen? That's right. The cholesterol might now diffuse out of the lipoprotein and appear in the plasma membrane of this cell, whatever it happens to be, altering membrane fluidity. When it alters membrane fluidity, the transporters receptors will not work, and then the cell will not work, the organ will not work, the body will not work, and then the person comes down with a terminal case of the DIDs. So the body says, you know, I want to live. And if the body wants to live, the body has to deal with this issue. How does the body deal with this issue? And the answer is it's going to take that cholesterol, put a fatty acid onto that hydroxy group to make a cholesterol ester. And by doing so, the cholesterol esters now are found in the interior. Cholesterol esters are found in the interior. And now comes the answer to the why are you putting the fatty acid onto the cholesterol? And the answer is to trap the cholesterol in the lipoprotein. So it will not diffuse out of the lipoprotein, go into the plasma membranes of cells when they collide, and therefore it'll be trapped in the interior. It will not diffuse out of the lipoprotein. That answers the why you're putting the fatty acid onto the cholesterol. Easy explanation. That's why they trap it in the lipoprotein. Here, but now the name of the enzyme in the bloodstream that puts the fatty acid onto the cholesterol, it's an important enzyme called, so the reaction has to take place is, here we have cholesterol, we put a fatty acid onto it to make a cholesterol ester. And the name of the enzyme that does that is an enzyme called ALCAT. So ALCAT puts the fatty, again, ALCAT is an enzyme made by the liver and secreted into the bloodstream. Here, so ALCAT carries out that reaction. But now, why do you have to transfer to the HDL to put the fatty acid on? Because HDL is remember, ALCAT has to be turned on. Has to be turned on by a apoprotein called ApoA. Remember the previous slide? ApoA turns on ALCAT. And it turns out the only place in the body you have ApoA is on the surface of HDL. So here's the ApoA on the surface of HDL to turn on ALCAT to make cholesterol esters. And then once the cholesterol esters are formed, then HDL will give cholesterol esters back to the IDL particle that goes on here. Amazing biochemistry that goes on here. Now, if I can make an analogy, here. Suppose I am HDL and on my surface is ApoA. And now I'm circulating in somebody's bloodstream right now. So as I circulate, when I encounter IDL, IDL is going to give me its cholesterol. And then IDL goes away. And now as I continue to circulate with cholesterol, my ApoA on my surface is looking for ALCAT. When ALCAT comes circulating by, my ApoA is going to say to the ALCAT, "Hey, ALCAT! Hey, ALCAT, you over there. Yeah, you. I'm talking to you. Come here a minute, ALCAT. Come here." And sure enough, ALCAT then docks with the ApoA on the surface of HDL. ALCAT will then put the fatty acid onto the cholesterol. And then after it does so, ALCAT will say to ApoA, "Am I done?" And ApoA will say, "Yeah, get out of here." And ALCAT then leaves. So, but now I have cholesterol esters in me, HDL. And as I continue to circulate, when I encounter IDL again, I'm going to say to IDL, "Hey, IDL, come here a minute. I got something for you. I did you a favor, IDL. And here is, remember that cholesterol you gave me? I put a fatty acid onto it for you. Here, have the cholesterol ester back." And it gives the cholesterol ester back to the circulating IDL. Remember, this is all as blood circulates. Incredible biochemistry going on. This is going on in your bloodstream right now, how the cholesterol is going back and forth from these different lipoproteins. Amazing biochemistry that goes on here. All right, so this explains the why, when, where, how, and blah, 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 all understandable. It's not easy. Don't get me wrong, it's not easy, but it is understandable if you think about it. If you try to memorize this, good luck. But if you think about it, it starts to make sense. Now, to finish off this slide, because we have more biochemistry to talk about. So the IDL, two fates. One is to be removed out of circulation by the receptor in the liver, but the other fate of IDL is when it encounters HDL again, it's going to give to the HDL its ApoE. And when it gives up its ApoE, then IDL at that point there converts to what's known as the notorious LDL particle. And what is LDL? It's much smaller in size, but it's mostly cholesterol esters in the interior. So it's mostly cholesterol esters Very little triglycerides, mostly cholesterol esters, milligram amounts. And on the surface is a single copy of ApoB100, not multiple copies, a single copy of ApoB100 on the surface here. Here. And now it's important to understand that the ApoB100 on the surface is a single copy, not multiple copies. That has everything to do with pathology in a little bit. You must understand one copy, not multiple copies. If you had multiple copies of B100 on the surface of LDL, there would be no pathology. But it's a single copy. I'll explain that issue a little bit later here. All right, now the obvious question next is, well, what's the fate of the LDL particle? That's coming up next. Now, before we get to that, what I want to do is to help you out and summarize a few items here. By looking at the slide, what it's telling you is in the human body, there are 6 lipoproteins, 6 of them total. 3 are made by an organ and 3 are degradation products. So here it comes. The 3 of the 6 lipoproteins that are made by an organ are, pay attention now, and they are chylomicrons made by the intestine, VLDL made by the liver, and HDL made primarily by the liver. The intestine can make a little bit of HDL, but most of it's made by the liver here. So 3 are made. Chylomicrons, VLDL, and HDL are made by an organ. And 3 are degradation products. And they are, here it comes, chylomicron remnants, a degradation product of chylomicrons, IDL, a degradation product of VLDL, and lastly, LDL, low density lipoprotein, a degradation product of IDL. 3 are made and 3 are degradation products. All you got to do is look at the figure. 3 are made and 3 are degradation products. Here. Now, another thing I want to point out is, now remember earlier this week I said to you, there are 3 laboratory values. You should know the number, what's normal, without looking at a chart, because you save time. One of them was a blood glucose concentration. Now, here's a second one. In my opinion, you should know the blood cholesterol values. What's normal without looking at a chart. And that cholesterol number should be 200 milligrams per deciliter or less. 200 milligrams per deciliter or less is considered normal blood cholesterol. Blood cholesterol here. And so the total cholesterol in the blood should be 200 milligrams per deciliter. Now, when you do a lipid profile, and by the way, you should do a lipid profile of yourself. If you have never had a lipid profile done, because you're going to tell patients, I want to see your lipid profile, but you should have it done of yourself first. If you never had a lipid profile of yourself, what are you waiting for? What are you waiting for? Are you waiting for it to be too late and then you have a heart attack and you get collapsed on the ground? At an early time of your life, like you are right now. Right now, you're a young person. While you're a young person, you should have a lipid profile done. Get a total cholesterol. You can also measure HDL cholesterol. You can also measure LDL cholesterol, and you should have those values. And then every year, have those values done again. Because as we'll talk about a little bit later at the end of the chapter, as you and I get older, your cholesterol number may have a tendency to start creeping up, and you don't want to wait until it's too late. You know, when I was your age, I had no problem with cholesterol. I can go to McDonald's. As long as they have a 2-for-1 special, I would buy 2 Big Macs and chug them down, things along that line. I had no problem with cholesterol. But as time goes on, as I started to get older, in many individuals, not all, but many individuals, the cholesterol number may start to creep up, and explain why a little bit later. And you don't want to wait until it's too late. So, for all the things I talk about here, don't worry about the exam. Forget about it. It's your life that's important. Have your cholesterol test done. Now, some people are going to say, "Oh, but you're a young person. We need to have it done." Hey, it's your life, not theirs. Have it done it, and every year monitor the total cholesterol, and total cholesterol and LDL cholesterol and HDL cholesterol. By the way, in a lipid profile, you will not find IDL cholesterol because IDL is a transitory particle. It's either removed or converts to the LDL particle. So, but anyway, you should have a profile done here. All right, now, because if a cholesterol number is in the low 200s, that's telling you it's not a genetic problem. It's likely a lifestyle problem. You gotta change your lifestyle if the number is in the low 200s. You might be eating too much red meat or other things along that line. So if it's in a low 200s range, we'll talk about in a little bit later things that can be done. If it's 250 or greater, it's telling you it's a genetic problem. But again, there's great pharmacology. In contrast to osteoporosis, in which the pharmacology is pretty bad, with cholesterol, there's great pharmacology, great drugs out there. And we'll talk about again in this chapter here. All right, so the next thing to talk about is what's the fate of LDL? Now, first of all, what does LDL look like? Now, this is what LDL is believed to look like, not in your book. But again, it's a particle with all inside the particle are all those cholesterol esters here. So the purple color or whatever color that is, that's cholesterol. That white line, that's the fatty acid. So cholesterol esters are on the interior, and on the surface is anything polar, like a little bit of phospholipids, a little bit of unesterified cholesterol would be on the surface. And remember, a single copy of ApoB100 on the surface here. Here. Now, by the way, if LDL looks like that in your monitor or your screen in your computer right now, if cholesterol was that size, what would VLDL look like in terms of size? VLDL will be the size of your room right now. Whatever room you're in right now, VLDL will be that size because it's mostly triglycerides. But now just to use your imagination. So in VLDL, the size of your room with all those triglycerides and cholesterol, what happens is when it acquires ApoC from HDL, ApoC turns on lipoprotein lipase, and triglycerides are being removed. Triglycerides are being removed. The VLDL is shrinking. And somewhere during the shrinking process, cholesterol is then being transferred to the HDL, shrinking. More triglycerides are removed. More cholesterol is being transferred to HDL, shrinking. Eventually gives off ApoC back to the HDL, shrinking. And then HDL will give me cholesterol esters, shrinking. Then ApoE is given back to the HDL, shrinking, shrinks all the way down to this thing right in front of you on the monitor right now. Amazing biochemistry that goes on there. How the VLDL shrinks all the way down to the LDL particle. Incredible biochemistry. All right, now what's the fate of the LDL particle here? Now that's coming up on the last slide here in biochemistry of cholesterol. Then the next number of slides is the pathology. All right, now, and by the way, this slide, something about the slide was on a national board exam according to a student. They were asking about how does a cell regulate how much cholesterol is in it? Cholesterol homeostasis. So again, pay attention to the biochemistry, and I'll explain the issue of cholesterol here. All right, now to orient ourselves, suppose that horizontal purple line represents a plasma membrane of many cells like hepatocytes. And that red circle represents the LDL particle with cholesterol esters in it. And on the surface is ApoB100. Now that blue rectangle you see there on the plasma membrane of cells, suppose that represents the famous LDL receptor. Now, for the LDL to come out of the blood into the cell, what happens is LDL docks with the LDL receptor. Now, let me stress, what is the LDL receptor seen on the LDL? It sees the single copy of B100, not the rest of the LDL. The receptor recognizes that B100 protein on the surface of the LDL particle here. That's what's being recognized by the receptor. Now, remember from histology, this is occurring in not on the surface of the entire cell, but only regions of the cell surface where they're clathrin-coated pits. Again, that's all cell biology, histology, integration. So LDL docks with the LDL receptor in the clathrin-coated pit regions of the membrane, and then endocytosis occurs. And then remember that the low pH of the endosome then dissociates the receptor from the LDL. The LDL receptor then recycles back to the surface to bring in more LDL. Here. And now, importantly, let me just say something here, because this has a lot to do with pharmacology a little bit later. Every time the LDL receptor recycles back to the surface, you lose a few percent. The LDL receptor gets degraded, just a few percent. And the few percent that's lost by degradation is replaced at the gene level. So you lose a few percent when it recycles, but you remake it at the gene level. Please keep that in mind. That'll be important in pharmacology before the slide is over. All right, so when the receptor recycles back to the surface, what happens to the endosome? Well, the endosome then fuses with lysosomes, and then lysosomes then break down the LDL. So all the ApoB100 is broken down to amino acids, but also in lysosomes, there's an enzyme called a lysosomal esterase that'll cleave off the fatty acid from the incoming cholesterol esters, and now we have cholesterol coming into the cell. It came in as a cholesterol ester, but you hack off the fatty acid, and now we have cholesterol coming into the cell. All right, now this cell says, okay, maybe we want some cholesterol, but we don't want to be flooded with cholesterol. Why? Because if there's a lot of cholesterol coming into the cell, that cholesterol can get in the membranes of the organelles, altering membrane fluidity of mitochondria and nucleus and so on. So a cell says, okay, maybe we want some, but we don't want to be flooded with cholesterol. That would be bad. So how does a cell regulate how much cholesterol is in it? Well, there are three ways a cell can regulate how much cholesterol is in it. The first of the three ways is shown here on the right side. Now, as we'll talk about at the end of the chapter, that you and I can make cholesterol by eating glucose. Here, sugar. So what happens is multiple glucose generate multiple acetyl-CoA, and then in cholesterol synthesis at the end of the chapter, there is a mega enzyme cholesterol synthesis. Don't worry about this enzyme now, but I will give this enzyme all the honor, all the praise, and all the glory it deserves at the end of the chapter. But the idea is, so this enzyme, the top enzyme of cholesterol synthesis. Now, as I mentioned to you earlier, cholesterol synthesis is expensive. You don't have to know this number, but it takes something like 293 ATP equivalents to make one cholesterol. So the idea is this, a cell has a choice. It can either make its own cholesterol, which is very expensive, or it can get cholesterol free from the bloodstream, from the environment. So if it has a choice, who do you think it's going to choose? That's right. It's going to go for the handout. So the incoming cholesterol will then inhibit the HMG-CoA reductase. Why make it if you can get it for free? So inhibit cholesterol synthesis. All makes sense. All right. Now, that's the first of three things incoming cholesterol does. A second of three things that cholesterol does when it comes into a cell is, when there's a large lot there, the cholesterol can then activate an intracellular enzyme called ACAT. Now, what does ACAT do? What ACAT does is, inside the cell, it'll take the cholesterol and put a fatty acid onto it to make a cholesterol ester. And once it does so, it then throws the cholesterol ester into a closet, into a vesicle. In other words, it gets it out of the way so it doesn't cause havoc to the cell. So again, you activate the ACAT enzyme, then it puts the fatty acid onto the cholesterol, and then you throw it in the closet until you need it. Get it out of the way so it doesn't cause havoc. Now, it says storage here, but you're not storing cholesterol esters like a stored triglyceride. It's simply you get the cholesterol out of the way, throw it in some vesicle or whatever, so it doesn't cause havoc to the cell. Now, let me stress here. Don't confuse ACAT with LCAT. Both enzymes make cholesterol esters, but the locations are different and the functions are different. So I'll say it twice in case you miss it the first time. ACAT is an intracellular enzyme for the purpose of making cholesterol esters for storage, getting out of the way. ALCAT is an extracellular enzyme in the bloodstream for the purpose of making cholesterol esters to trap the cholesterol in lipoproteins. So one more time, don't confuse ALCAT and ACAT. Both of them make cholesterol esters, but the locations are different. One's inside cells, one's in the bloodstream. And the functions are different. One's to make cholesterol esters for storage, getting it out of the way, and the other is to trap the cholesterol in lipoproteins. So that's the second thing that incoming cholesterol does. And now the third thing incoming cholesterol does is it inhibits the gene expression for the LDL receptor here. So when cholesterol comes in, it represses the LDL receptor gene expression here. Now, why is that important? Because now remember, as I mentioned earlier, when the LDL receptor recycles back to the surface to bring in more LDL, you lose a few percent. That's by just protein degradation. That's replaced at the gene level here. But now, if there's a lot of cholesterol coming to the cell, then you're going to inhibit the gene for the LDL receptor. So therefore, every time you lose a few percent, lose a few percent, lose a few percent, not being replaced, lose a few percent, lose a few percent, not being replaced. Eventually, there'll be no more LDL receptor on the surface because it's not being remade at the gene level. This is called downregulation. This is called downregulation of the LDL receptor. By inhibiting the gene expression, then every time you lose a few percent of the receptor, not being remade, eventually as time goes on, there'll be no more receptor on the surface. Downregulation of the receptor here. All right, now on the lower left side of this figure, I'm going to restate what I just said. You don't have to write it down because what I just said is shown there in the purple circles. But what I just said is this. If there's a lot of cholesterol coming into the cell, a cell says, I don't want to be flooded with that cholesterol. So the incoming cholesterol then will inhibit the HMG-CoA reductase enzyme. Therefore, you will not synthesize cholesterol in the cell. Another thing incoming cholesterol does, it activates the ACAT enzyme to put a fatty acid on, to throw it in the closet for storage. Get it out of the way until you need it. And then the third thing incoming cholesterol does is that it inhibits the LDL receptor gene. Therefore, there will be no more LDL receptor on the surface. Therefore, you will not take up the LDL. Cholesterol will not come into the cell. Now, on the left side, that's when the cell has a lot of cholesterol in it. Now, suppose a cell wants cholesterol, needs cholesterol. Again, you don't have to write this down. I will, but it's shown there in the purple circles. Now, suppose there's not very much cholesterol in the cell, and the cell wants cholesterol. Then the HMG-CoA reductase enzyme no longer being inhibited, no longer being repressed. It's now free to make cholesterol, to synthesize cholesterol. And secondly, the ACAT enzyme no longer being turned on, therefore you will not store it, the cholesterol esters. And then thirdly, the LDL receptor gene no longer being repressed. Then the LDL receptor gene can be turned on to make the LDL receptor protein. We put onto the surface to bring in more LDL, and now more cholesterol come into the cell. So on the left side, to summarize, on the left side, that's when too much cholesterol is in the cell. On the right side is when too little cholesterol is in the cell. Now, when things happen normally as they should, eventually these two things, too much, too little, eventually they'll bounce off each other. And that's called cholesterol homeostasis. And that determines your cholesterol number. When it's too much or too little, eventually it balances off here. Again, this is on the boards. They're asking about cholesterol homeostasis. Again, balancing off each other here. Here. Now we're almost done with the biochemistry here. Just a couple more comments here. But this also explains, suppose you get a lipid profile done of yourself, and suppose the cholesterol number is about 250. Again, it's definitely on the high side, 250. So if you go on to a cholesterol-free diet, no cholesterol in your diet, do not expect your cholesterol number to drop significantly. It might drop down to 245, 240. It's not going to drop very much if you go onto a cholesterol-free diet. Why not? Because if you don't eat it, the body will make it. So that's the idea. And by the way, if your cholesterol number is high, you should not eat it though. But don't expect your number to drop significantly, but you should not eat it. Why? Because we'll talk about in a little bit, before the day is over. Once you have cholesterol in your body, you and I are going to have a hard time getting rid of it. So you should not eat it if the cholesterol number is on the high side, but don't expect that number to drop significantly here. All right, now that's the biochemistry here. But now comes the integration with pharm and the integration with a lot of pathology coming up here. And now remember earlier when we were talking about that the LDL receptor gene is degraded. The gene not degraded, the protein is degraded, and you lose a few percent every time, replaced at the gene level. Remember that? That little bit of information, that information is the target of pharmacology. Now, later on at the end of the chapter, we'll talk about other drugs in pharmacology to treat hypercholesterolemia, like the statin drugs and so on. But there's another drug recently on the market. And I've seen commercials on TV. What they say is that if statin drugs are not working as they should, or if they cause too much rhabdomyolysis, they say you can take this drug to help lower serum triglycerides. And what is that drug in pharmacology? Anybody know that drug in pharmacology? And what it does so, it has something to do with the LDL receptor gene degradation. And that drug is, that's right, that's right, it is a monoclonal antibody, commercially known as Repatha. And what does Repatha do? It's a monoclonal antibody, meaning it's going to be expensive. And because there's commercials on TV, that means it's going to be expensive. Somebody has to pay for the commercial. And so this expensive drug, but what it does is it binds to those proteases, prevents the proteases from degrading the LDL receptor. So therefore, the LDL receptor is not being degraded. It'll be put onto the surface to bring more LDL cholesterol out of the blood into the cell. Get it out of the blood into the cell. So that is the biochemical basis of the pharmacology. Now the other in pathology, what's going to happen? Just use your imagination here. What's going to happen when the gene for the LDL receptor is defective? What's going to happen to that the LDL? Well, what happens is in pathology, that's called type 2A hyperlipidemia, also called familial hypercholesterolemia here. Now, just think of the consequence here. Think of the consequence. When that gene is mutated, that means on the surfaces of the cells, there's no functional LDL receptor. And then what happens to all the LDL in the blood? It stays in the blood. Hypercholesterolemia. And if you think that's bad, that alone is bad. Then if you think that's bad, this cell multiplied by many, many cells in the body think they're starving for cholesterol. Even though there's plenty of cholesterol in the bloodstream, cholesterol cannot come into the cell. The cell starts to starve for cholesterol and now starts to make more and more and more cholesterol, even though there's plenty of cholesterol in the bloodstream, contributing to the hypercholesterolemia. And as the months, as the years go by, you can imagine the amount of cholesterol in the body here. So that's why at a very early age, you better get your cholesterol test done. Know your number. Know what it is at an early time in your life. All right, now let's talk about LDL. When it stays in the blood, for whatever the reason is, either because of genetic problems or lifestyle problems, suppose there's a lot of cholesterol in the blood. So now this is going to be a lot of pathology here. here. And so now what this slide is about is to explain what good cholesterol is versus bad cholesterol. You know, your friends or your relatives, well, as since you're a physician or going to be a physician, they're all going to come up to you and say, hey, I heard an LDL is bad. I heard HDL is good. Good cholesterol. Could you verbalize a simple, coherent explanation? That's what this slide is all about, to help you try and explain what's going on with this cholesterol business. All right. Now, when you hear the word bad cholesterol, everybody thinks that's LDL. Right at the beginning, let me tell you, LDL, despite what everybody says, LDL is not bad for you. LDL is not bad. But what is bad for you is this. Remember, in the bloodstream are all those free radicals, superoxide, because you're breathing in oxygen and so on. There's a lot of free radicals in the bloodstream, always looking for electrons. So what can happen is those free radicals in the bloodstream, they can take, what else is in the bloodstream? LDL. And so what happens is those free radicals can take the electrons from that single copy of B100. And by taking electrons from a single copy of B100, that B100 now becomes oxidized and now is damaged, and that product is now called oxidized LDL. That is the bad player, not LDL. It's oxidized LDL. That's the bad player. But here, so now what's the problem? The problem is because that single copy of B100 is damaged by oxidation. That means the LDL receptor will no longer recognize it. So if there's any LDL receptor around on the membranes of your cells, it no longer recognizes the oxidized LDL because the ApoB100 is damaged. This explains, so the LDL will not come out of the blood. Oxidized LDL will not come out of the blood. This explains why a single copy is so important to understand. If you had multiple copies of B100 on the surfaces of LDL, if one got damaged by free radicals, no big deal. The LDL receptor would see one of the other ApoB100s. But the fact there's a single copy of B100 on the surface, when it gets damaged, then the entire oxidized LDL then looks foreign to the human body. When it looks foreign to the human body, here come the macrophages. So what happens is the macrophage, and again, a lot of this is going to go on in the subendothelium. So what happens is the macrophages then see this foreign substance circulating in somebody's bloodstream, and sure enough, the macrophages will then engulf and take up the oxidized LDL, will now come into the macrophage. And then the oxidized LDL comes into the macrophage, and then by endocytosis as well. And then the endosome then fuses with lysosomes, and then you're breaking down the oxidized LDL all the way down to cholesterol in the macrophage here. All right, now importantly, how does in immunology, how does the oxidized LDL get into the macrophage? By what receptor? And by the way, on this slide, there are going to be two more receptors. One of them was on the boards. I forget which one. But what receptor does a macrophage use to take up the oxidized LDL? Does anybody know? Which scavenger receptor? And that is scavenger receptor A. Scavenger receptor A is this receptor on the surface of macrophages to which the oxidized LDL binds to and comes into the macrophage. Importantly, what is the difference between the scavenger receptor A and the normal LDL receptor? Besides location, what's the difference between scavenger receptor A and the LDL receptor? And the answer is the scavenger receptor is not down-regulated by incoming cholesterol. The LDL receptor is, but the scavenger receptor A is not. So what? So what does that mean? So what's the significance of that? The significance is easy to understand. The significance is the macrophage continues to eat and eat and eat and eat oxidized LDL just like a big fat pig. It doesn't know how to stop eating. It continues to eat and eat, not done regulated, eat and eat oxidized LDL, all this incoming cholesterol coming in, oxidized. So then the macrophage becomes so bloated with all that incoming cholesterol that in pathology, the cholesterol bloated macrophage is now known as a foam cell. A foam cell is a cholesterol bloated macrophage. Again, a lot of this is going on in the subendothelium. So what's the problem with the foam cell? It's now useless as a macrophage, and eventually the foam cell will die. Like when any cell dies, it spills its guts into the environment. And all the sodium and all the glucose in the foam cell just dissolve in the bloodstream, no big deal. But all the cholesterol that's in the foam cell will then precipitate out because it doesn't like water environment of the bloodstream. So that's what all those yellow dots you see here is representing cholesterol that now precipitates out when the foam cells dies. And as you know, in pathology, that's now the beginning of the classic atherosclerotic plaque. Monocytes come in, you get the inflammation, the CRP protein is then released and so on. And beginning the plaque being formed. And as you can imagine, as time goes on, the plaque is getting bigger and bigger and bigger here as the months and years go by. Here. Now, on the right side, that's just a cartoon of the plaque, but this is the real life situation. So what you're looking at right here, here is the plaque right here indicated by the blue arrow. Underneath in yellow is all that cholesterol that precipitated out. And here is a blood vessel where things are circulating by. Now, as you can imagine, as time goes on, that plaque is getting bigger and bigger and pushing up. And a very dangerous situation that goes on there. All right, now here, so all these, so now when the blood circulates by that plaque, what happens is, no one likes cholesterol. You ask your friends and relatives, you like cholesterol? Ooh, cholesterol, I don't like cholesterol. And same thing with the molecules in your bloodstream. They don't like cholesterol either. When albumin comes circulating by, albumin laughs at the cholesterol precipitated. Look at that thing. Laughed all he's sitting down. Nobody likes it. And not only that, hemoglobin comes circulating by and flips it to bird and things along that line. Nobody likes cholesterol except when HDL comes circulating by. When HDL sees that precipitated cholesterol, it feels sorry for it. And HDL says to that cholesterol, hey, cholesterol, what are you doing all by yourself? Come circulate with me. And sure enough, the HDL will now take up that cholesterol that's precipitated out. So the cholesterol now come into the HDL. And what's the HDL going to do with that cholesterol? As we talked about earlier, what it can do is put a fatty acid onto it to make a cholesterol ester and give it back to circulating IDL. That's true. But in addition, HDL can then bind to a receptor on the liver only. And that receptor is called the scavenger receptor B1. That's the other receptor. That's the one they found on the liver cells to which HDL docks. And when HDL docks, it deposits the cholesterol in it back into the liver. Remember, cholesterol came from the liver, but now going back to the liver from HDL. It's sometimes called reverse cholesterol transport. You're transporting the cholesterol back to the liver here. This is why HDL is considered good cholesterol, because the higher the level of cholesterol, the higher the level of HDL you have, if the number is 40 milligrams per deciliter, it's not very good. If it's in the 50s range, it's much, much better. If it's in the 60s range, 70s range, that's great. The higher the level of HDL, the better to scavenge the cholesterol. Keep it moving. Keep it from precipitating out. So the HDL will scavenge it and try to do something with it. Bring it back to the liver, in the reverse cholesterol transport, or give it to the IDL as a cholesterol ester. But after all, you're going to say, what is the liver going to do with that cholesterol? Well, the liver will then reform into a VLDL, which converts into IDL. Then IDL will convert to LDL. So anyway, that's why HDL is considered good. Because the higher the level of HDL, back off here a minute, the higher the level of HDL, the better to scavenge the cholesterol. But here you're saying all you're doing is making more LDL. That's true. But LDL is not bad for you. As long as the LDL is circulating, as long as cholesterol is circulating in the LDL, it's not a problem. But why is the LDL considered bad? It's because of this arrow. The greater the amount of LDL you have, if that number is 100 milligrams per deciliter of LDL cholesterol, it's not good. So if it's greater than 100 milligrams per deciliter, the higher the LDL, the greater the probability of generating oxidized LDL, because this is a non-enzymatic reaction here. By mass action, the more LDL you have, that means you're going to have more oxidized LDL here. So that's why LDL is considered bad, even though technically it's not. And the more oxidized LDL you have, the better the probability of generating foam cells and blah, 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 blah. We talked about a few minutes ago here. So that's why LDL is considered bad, although technically it's not. Now, if the cholesterol number is in the low 200s, it means it's a lifestyle problem, not a genetic problem. You got to change your lifestyle. Stop eating so much trans fat and saturated fat, and stop eating so much red meat. You should be exercising, things along that line. High fiber diet is good. We'll talk more about high fiber diets after the break. But another thing to be done is take some antioxidants to try to slow down this arrow here. But if you take antioxidants like vitamin E or resveratrol or whatever, you're trying to slow down that arrow, preventing LDL from converting to oxidized LDL. So that's why, and there's things that can be done. That's why you should know your cholesterol number, because if it's in the low 200s, you got to do something about your lifestyle here. So that's the idea. Okay, right now I'm checking the time. It's almost the bottom of the hour. So what we're going to do is take our last 10-minute break and then come back. And then we'll get into what happens if the cholesterol number is in the 250s or greater. It tells you that's a genetic problem. So we'll talk about some genetic diseases associated with lipids. Then after that, then we'll talk about cholesterol synthesis and some pharmacology, pathology, physiology there. And then we're done with the chapter. And then we'll get into the next chapter after that. Okay, 10-minute break, and see you shortly. And make sure I turn off the microphone this time. Okay, everybody, welcome back for this last hour. So again, if the cholesterol number is in the low 200s, that tells you 205, 210. It tells you it's a lifestyle problem. You got to change the lifestyle here. But if it's 250 or greater, likely it's going to be some genetic problem, especially if it's in the 300s range, likely a genetic problem here. All right, now in terms of genetic diseases associated with lipids, these are the two most important ones, type 1, type 2A, hyperlipidemia. Now, when Dr. Brome may or may not cover the other ones, type 3 and type 4, but these are the two most important hyperlipidemias for the boards, type 1 and type 2A. Here, these are genetic disorders. Now, type 1 is a gene defect. Some people have a gene defect of lipoprotein lipase, the enzyme we talked about earlier. Other people in the world have a gene defect in ApoC2. So either gene being mutated, then you end up with the same disease called type 1 hyperlipidemia. So these genes may be on different chromosomes or whatever, but you end up with the same disease. In genetics, that's called locus. It's called locus heterogeneity. The locations are different. Either gene being mutated, you end up with the same disease. So in genetics, that's called locus heterogeneity here. All right. Now, when either protein is not working as it should, then triglycerides cannot be broken down in chylomicrons. Triglycerides fatty acids will never enter the cell. So in the bloodstream, you expect to see elevated triglycerides in the bloodstream. Here, hypertriglyceridemia, or you might see it called hyperchylomicronemia. So again, different names for the same disorder. Type 1 is also called hypertriglyceridemia, or hyperchylomicronemia is type 1 here. Now, in terms of pathology, yes, they have fatty livers. Yes, they have acute pancreatitis. Yes, they have abdominal pain after a fatty meal. But a lot of things cause those things. Alcoholism causes fatty liver. Removal of the gallbladder causes abdominal pain after a fatty meal. So those are characteristics, yes. But what is the main hallmark? And there it is, the red-orange eruptive xanthomas. You see that phrase, or they actually show you the red-orange eruptive xanthomas. These are little pimples that can erupt at any time. And again, that would be characteristic of hallmark of type 1 hyperlipidemia here. Look at the triglyceride level in this patient, 1,600. The normal triglyceride levels is about 100 milligrams per deciliter. Clearly, this is a genetic problem, not a lifestyle problem in type 1 in this particular patient here. Now, type 2A, an even more important disease for board exams, that's where the LDL receptor gene is. Oh, by the way, I forgot to mention here, you can also, in type 1 hyperlipidemia, also in blood values, have elevated VLDL. Because remember, VLDL also carries triglycerides. VLDL also has ApoC to turn on lipoprotein lipase. So in type 1 hyperlipidemia, yes, chylomicrons will be elevated if they eat a high fat meal. But if a person eats a high carbohydrate meal, then you expect to have elevated triglycerides in VLDL coming from the liver. Again, nothing strange, nothing unusual with that information. Now, type 2A is a gene defect of the LDL receptor. Now, remember from Dr. Hardin's lectures, type 2A is autosomal dominant mode of inheritance. And so it's so in the homozygous situation in which both genes, the one inherited from the mother and the father, both of them are mutated. It's very rare. 1 in a million people have that. But there, the cholesterol number is so high, where there's no LDL receptor on the surface. The LDL in the bloodstream is so high, carrying all that cholesterol, that the blood cholesterol number might be 700 milligrams per deciliter compared to the normal 200. 700 milligrams per deciliter. There's so much cholesterol in the bloodstream, clogging up with all those plaques and so on. That unfortunately, those children, they'll live for a short period of time, but usually die at an early age of a heart attack at age 5 or 6. So much cholesterol in the bloodstream here. In the homozygous situation, which is very rare, 1 in a million. But much more commonly is a heterozygous situation in which a person has a good gene and a bad gene. They still have pathology, but it's about 1 in 500 individuals have the heterozygous situation. And there, remember, with autosomal dominant in the heterozygous situation, it's a late onset. For 20 years, 30 years, 40 years, no problem, no pathology, but their cholesterol number might be 300 milligrams per deciliter, no problem for many, many years. But after about age 35, age 40, 45, 50, there's so much cholesterol in the body that they end up with those xanthomas of the Achilles tendon, the hallmark of that pathology here. And they have so much cholesterol in the body with all those xanthomas of the eye, the xanthalmas and so on, it's prime for a heart attack at age 45, 50, 55, something along that line. Late onset in the heterozygous situation, it takes a long time for the pathology to really show up here. But that's why you really need to get the cholesterol numbers done of patients at very early ages who might be susceptible to hypercholesterolemia here. Now, in addition, they can have by the way, it doesn't have to be xanthomas of the Achilles tendon. It could be xanthomas of the knuckles as well. Again, characteristic of type 2A. Now, in addition, another hallmark of the pathology is corneal arcus. Now, if I were to ask you right now, what is corneal arcus? You're all going to say it's like a little grayish arc around the cornea. So if you're in ophthalmology and some patient comes in your clinic and you examine the eye and then you have this grayish arc around the eye, you tell that patient, hey, you better go to a family practitioner right now or an internist and have your lipid profile done because there's a time bomb ready to go off in the body. Again, a very dangerous situation in the body, but these are characteristics of type 2A hyperlipidemia. Okay, again, type 3 and type 4, Dr. Barone may or may not talk about those. In my opinion, they're not as important compared to these two right here for U.S. boards here. All right, now the last thing in this chapter we want to cover is cholesterol synthesis. Now, in terms of the biochemistry of the pathway, the biochemistry really, in reality, is very difficult pathway. Lots of enzymes, lots of structures and things along that line. It is a very difficult pathway in real life. But for the boards, cholesterol synthesis is a piece of cake. It's a very simple pathway for the boards. Why? There's only one enzyme you need to know, and all the other enzymes, forget about it. So don't panic when we go to the next slide and talk about cholesterol synthesis. It's a very simple pathway. All right, now why is this a high yield figure? Because on this slide, integration with the 3 Ps. But let me do the biochemistry first. So now what happens is, as I mentioned earlier, cholesterol has 27 carbons. All 27 carbons come from multiple glucose. So multiple glucose generates multiple acetyl-CoA. 2 of them condense to form the 4-carbon acetoacetyl-CoA. More condensation generates the 6-carbon HMG-CoA. Now, once HMG-CoA is generated, it sees the top enzyme of the pathway, if not one of the top enzymes for the boards, mega enzyme for the boards called HMG-CoA reductase here. Now, this is the only enzyme you didn't know in this pathway here. Now, this enzyme, now, why is it so important? We'll talk about why in just a few minutes, but I wonder why. But why is this enzyme? But now, what does the enzyme do? In terms of biochemistry, it tells you what it does just by the surname, reductase. So what do you think this enzyme needs to work, by the way? Not shown on the slide. What do you think the enzyme needs to work? Do you think it needs biotin? Do you think it needs vitamin D? What do you think this enzyme needs to work? HMG-CoA reductase. You would imagine it, because it's a reductase, it's going to put electrons back into the substrate to make a product. Therefore, it's likely to require NADPH. NADPH. So it needs NADPH to carry out that arrow. That's not shown here. That converts hydrogen peroxide, HMG-CoA to mevalonic acid here. Needs NADPH. Anyway, let's proceed. We'll come back to this enzyme in a few minutes. So it generates the 6-carbon mevalonic acid. Then after a few steps you don't have to know, makes the 15-carbon farnesyl pyrophosphate. Now, most of the farnesyl pyrophosphate is used to make cholesterol, but it's important to know that some of this farnesyl pyrophosphate can be used to make other molecules in the cell, namely coenzyme Q. That's the most important one you need to know. Yes, it can make dolichol pyrophosphate. Dr. Harden may have talked about that or not. Not a big deal, but you should know it's used to make coenzyme Q. That will be important in pharmacology in a few minutes here. Now, if it's not used to make coenzyme Q, the vast bulk of farnesyl pyrophosphate is used to make cholesterol. So what happens is 2 molecules of the 15-carbon farnesyl pyrophosphate come together in some sort of ancestral marriage to form the 30-carbon squalene. Then squalene converts to lanosterol. And then after a few more steps you don't have to know, lanosterol then converts to cholesterol. So there it is. That's cholesterol synthesis, a piece of cake. Very simple pathway for the boards. Now, once cholesterol is made or in a cell, or you get it from a diet from in the cell, what can happen to that cholesterol? Remember, I touched on this earlier. You cannot burn the rings of cholesterol. You can put the rings together, and this pathway is shown in front of you. But once the cholesterol rings are put together, there is no enzyme in the body capable of cleaving any of the rings of cholesterol. We can add to the rings, subtract from the rings, but we cannot cleave the rings of cholesterol. Therefore, you cannot burn it to CO2 and water. So therefore, what can the body do with the cholesterol? Well, one thing the body can do with the cholesterol, if you cannot burn it, one thing is it can take the cholesterol and make the steroid hormones here. All right, here comes the question for you. This arrow, cholesterol making steroid hormones, is that a major arrow in amounts or a minor arrow? Is that a major arrow or minor arrow? Cholesterol making the steroid hormones, major or minor arrow? And the answer is, it is a minor arrow. After all, how much testosterone do you need? You don't need very much testosterone. So it's major in significance, but minor in amounts. Remember, you have milligram amounts of cholesterol. And the steroid hormones are needed in microgram or nanogram amounts, 1,000 or 1,000,000 times less. So the amount of cholesterol that you always hear about in other courses going on to make steroid hormones, yes, that's significant in terms of what it's doing. But in terms of amounts, it's a minor arrow. Next arrow. Cholesterol making bile acids. Is that a major arrow in amounts or a minor arrow? Major or minor arrow? Cholesterol making bile acids. And that is a minor arrow. Now, why is that a minor arrow? Again, now this integrates with GI physiology or anatomy. Remember, the bile acids are made by the liver. And in the liver, there is the main enzyme, the top gun of bile acid synthesis is an enzyme called 7-alpha hydroxylase. So 7-alpha hydroxylase is the top gun in bile acid synthesis. But remember, when the bile acids are made by the liver, by this enzyme, the bile acids then go get into the intestine from the bile duct. They do their thing of emulsification. And remember, the bile acids are recycled back to the liver. They get absorbed and in the mucosa, then go back to the liver. And when they go back to the liver, they inhibit the 7-alpha hydroxylase. And then they go back into the intestine, back to the liver, back to the intestine, back to the liver, 6 to 10 times a day. And in physiology or anatomy, that's called enterohepatic circulation of bile acids. They go back and forth, 6 to 10 times a day. And every time they go back to the liver, they inhibit the 7-alpha hydroxylase. So in me and you right now, That enzyme's not working very much. Slide. Next arrow, cholesterol making vitamin D, major or minor arrow? And you're all going to say it's a minor arrow. Why? Because after all, vitamin D is going to be used as a hormone. Don't need very much. One last arrow in me and you. Cholesterol being incorporated in plasma membranes of your cells. Is that a major arrow or is that a minor arrow? Major or minor arrow? Look again. It's a minor arrow. Now, why is it a minor arrow in me and you? Because you're not thinking. Because in me and you right now, we're not growing very much, right? You're not growing right now. Next week for your course next week, your brain's not going to be 5x in size, maybe in terms of content yet, but your brain's not going to be 5x in size next week. You are not growing. Most of our cells are in G0 phase. The cholesterol is already there in the membranes here because most of our cells are not growing here. So, but in children, this is a major arrow because all the cells are growing. They need lots of cholesterol and so on. That's why you never tell a child, I want to see your lipid profile. Unless, of course, they have a genetic problem. But in me and you, this is a minor arrow here. And now look at the bottom of the slide. That's why in an early age that you and I are, well, I was, that you are right now, you should get your lipid profile because notice here, once you become an adult, cholesterol has very little places to go. And so if you start eating a lot of eggs and all that cholesterol and so on, not paying attention, I'm telling you, cholesterol is going to be a problem individual if you're not paying attention because you cannot burn it to CO2 in water and it has very little places to go. That's why the cholesterol number may have a tendency to creep up as a person gets older because it has very little places to go. All right, now continuing on. Now that enzyme HMG-CoA reductase, why is that enzyme so important? Well, in physiology, it's important because in endocrinology, it's written here, but you would predict that glucagon turns off the enzyme by phosphorylation mechanisms. Insulin turns on the enzyme by dephosphorylation mechanisms, and also at the gene level. Allosterically, the enzyme is turned off allosterically. So this enzyme is regulated all three ways. At the allosteric level with cholesterol, at the phosphorylation, dephosphorylation level with these hormones, and genetically turned on by insulin. All three ways, how the body goes all out to only turn on the enzyme when you're in a time of plenty, a lot of carbohydrate around. And when you're starving or fasting, you shut down the enzyme because why waste energy making cholesterol when you're starving or when plenty of cholesterol already in the cell? Shut down the enzyme. It all makes sense. You don't have to look up in first aid. It's all logical. Now in pharmacology, here come the statin drugs. Now, you should know in pharmacology, there are 6 statin drugs that are commercially available. All of them work the same. Whether it's Lipitor, Zocor, Crestor, they all work the same. Now, the way they might do it is this. They might say to you, this is a Lineweaver-Burke plot of a very important enzyme in cholesterol synthesis in the black line called HMG-CoA reductase in the black line. And in the red line is HMG-CoA reductase in the presence of atorvastatin. What type of drug is atorvastatin in the red line? And you say, well, the Vmax, no change, the Km changed. That means all the statin drugs are, because the lines cross, all the statin drugs are competitive inhibitors. Yes, it's true. They all look like HMG-CoA, the substrate. So they're competitive inhibitors of HMG-CoA. All right, now another item to mention here in pathology and pharmacology. Because now if a patient comes in your clinic, you do a lipid profile, and you find out they have hypercholesterolemia. So then you prescribe a statin drug. Let's say 80 milligrams per day. And so that's a pretty high dose, 80 milligrams. So then you tell that patient, that after about a month or so, come back, let's get another lipid profile done to make sure the statin drug is working. But also we want to do liver function tests. And if there's any problem with the muscle problem, come back and see me right away. You're going to tell that patient. So now the question is why? Why is it that some, not all, but some patients, especially on the high dose statin, can end up with rhabdomyolysis, and also you got to do liver function tests? Why is that? And the reason is, here comes the biochemistry explains all that. If you inhibit this arrow with a statin drug, yes, you don't make cholesterol. That's the objective. But you also don't make farnesyl pyrophosphate. And farnesyl pyrophosphate is used to make coenzyme Q, also called ubiquinone. Where do you have coenzyme Q? In mitochondria. Where's a lot of mitochondria? In the liver and muscle cells here. So that's why you have to do liver function tests, as well as asking about rhabdomyolysis, because all the side effects of a high dose statin is the issue of can be explained by the biochemistry. Now, as a physician, then what are you going to? Then what would you do? Again, it's all going to be person specific. One thing you could do is try to lower the amount of the statin drug from 80 down to 40 or something, see if it works without having muscle pain. Another thing, to change the statin to another statin. Another thing can be done. Remember Repatha we talked about earlier? You can now have a low dose statin, 10 milligrams, and then supplement it with a Repatha drug. Try that type of thing. There'll be another class of drugs coming up. We'll talk about in a few minutes here. But in addition, another thing can be done if somebody has rhabdomyolysis is that person can go to the store and buy coenzyme Q. Because after all, as I mentioned earlier, coenzyme Q, CoQ10, same thing. It's a lipid, not a protein. It will survive digestion. And some individuals, they take CoQ, it'll be digested properly, it'll come and be absorbed properly, and hopefully in some individuals, it goes to where it's supposed to go, in mitochondria, in liver, and muscle cells. So it's definitely worth trying with this coenzyme Q supplementation if somebody has muscle pain. If it doesn't work, again, person specific, then try other things. And one of the other things that can be tried is another class of drugs. And this other class of drugs also begin with the letter S. They're not the statins, they're called the sequestrants. And an example of a sequestrant is cholestyramine. Now, what is cholestyramine? And in these sequestrants, what's the strategy of the biochemistry there? The strategy is cholestyramine is like it's a powder. It's like very tiny grains of sand. And on the periphery of the grains of sand, there are positive charges. Look at the word. Cholestyramine, amine, positive charges. So on the surfaces of the cholestyramine powder, there are positive charges. So when you eat this stuff, it comes into your intestine. You cannot break it down, and out the body it goes. Now, because they're positively charged, as you might imagine, anything negatively charged will bind to them. And what's negatively charged? Bile acids. So the bile acids, just like a magnet, the bile acids now will stick to the surface of the cholestyramines, and then out the body they go. So the bile acids are now going out the body. They're not being recycled back to the liver. So as a result, the 7-alpha-hydroxylase is no longer being inhibited. It's free to make free to make bile acids. And if it's going to make bile acids, what does it need? It needs cholesterol. So now the body's doing something with the body cholesterol. So in other words, this arrow that's normally a minor arrow now becomes a major arrow. You're doing something with the body cholesterol, converting it to bile acids, and out the body it goes. Kind of a weird strategy, but it works. It works here. Yeah. The problem with this type of diet the problem with this type of therapy is, would you like to eat powder for the rest of your life here, or eat grains of sand for the rest of your life? Because after all, the person next day is not going to feel any better. And if a person has an infectious disease, you give them a macrolide, the next day or two or three, they'll feel a lot better. But here, they're not going to feel better. It's working, but they're not going to feel physically better the next week or month. So therefore, a person may say, I don't want to take this pot, or I don't know if it's working or not. That's the problem with the sequestrants. Keeping the person on that regimen. But it's working, but it's just, you just don't know it. By the way, one last thing here. If your cholesterol number is in the low 200s, then another type of thing that can be done in terms of lifestyle is eating a high fiber diet. High fiber diet. And it's soluble fiber, not insoluble fiber, like lettuce. But if you eat soluble fiber, what happens is, like, for example, if you take some Metamucil, commercially known as Metamucil, the active ingredient in Metamucil is something called psyllium. Psyllium, P-S-Y-L-L-I-U-M. P-S-Y-L-L-I-U-M. What it is, some plant soluble fiber. That's the active ingredient in Metamucil and all these dietary things. And what happens is if you take some psyllium and put it into a glass and add some water, what happens is it forms like a big colloid. And what happens is when you eat this stuff, you cannot break it down, and out the body it goes. And for many, many years, psyllium was beneficial for humans because it helps to give rise to proper peristalsis. In other words, a person has constipation or whatever, it helps to get the flow, the peristalsis through the intestine and out the body. But they found out it had another benefit. In fact, a physician from my institution reported on this. Has another benefit, and that is when it goes to the intestine, out the body, it traps some of the bile acids. So it traps the bile acids and some of the cholesterol, and out the body they go. And there's good evidence for that, science evidence for that. It lowers total cholesterol by 5 to 10%. But 10% of 205 will get you down to an acceptable number. So that's the idea. So if it's worth trying a high fiber diet, with Metamucil or something cheaper than that. But again, the active ingredient is psyllium, a soluble fiber. Anyway, there it is. Now we're done with chapter 15, a high yield, chapter with all that cholesterol that you want to know for the boards. But importantly, even more importantly, for your career, you got to know what's going on there. Oh yeah. Okay. So now we're done with this slide, with this deck. Maggie, can you bring in the other deck? There we go. Thank you. So now this is our last deck, is chapter 16 through 18. So we got 4.5 hours. So we're just about where I want to be here. Okay. So now on this chapter 16 is the second of the two lipid chapters. This deals with the reverse because the previous chapter was synthesis and storage and stuff, but now we're talking about mobilization and catabolism, the breakdown and so on of lipids and so on. Again, let me just tell you, from now until I'm done tomorrow, every slide, high yield, high yield, high yield, high yield, every slide you can get questions on board exams about every slide. I'm going to talk about now until I'm done tomorrow. All right. Now the first thing I want to talk about here is lipid mobilization on page 238. 238. All right. Now, before I talk about the slide, how you mobilize triglycerides from the adipocytes. Before I talk about the slide, let me mention something here. This slide, the biochemistry of this slide is the basis of the Atkins diet. Okay, again, the Atkins diet was a diet about 10 to 15 years ago or so, and then it sort of faded out and then came back. Now it's something called the ketogenic diet or the keto diet. The basis of the diet is this slide. And you'll never understand that diet until you know the biochemistry first. So let's go through the biochemistry. Then we'll talk briefly about the ketogenic diet. Is it a good diet or not? You can make that decision. Anyway, let's talk about the biochemistry. Now, what we got over here on this slide, on the left side is the adipocyte. In the middle is the bloodstream. On the right side is the liver. Now, here are all these triglycerides in the adipocyte. Now, how they got there, that was chapter 15. All the stuff about chylomicrons and VLDL and lipoprotein lipase, blah, 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 blah. That was the previous chapter. But now these triglycerides, there comes a time when you need them. And what happens is when there's low levels of insulin, increased epinephrine, increased cortisol, not shown here, increased glucagon, what happens is they will turn on a major enzyme for triglyceride mobilization. An enzyme we talked briefly here and there over the last several days, hormone-sensitive lipase, will now turn on, and triglycerides break down to 3 fatty acids and glycerol. 3 fatty acids and, there's a pointer, 3 fatty acids and glycerol here. All right, now before we proceed, it brings up a couple items here about that enzyme, HSL. Now that enzyme exists in 2 forms. One form, like you see right there, highlighted in the red box, and the other form is the red box plus a phosphate group on, like you see right there. So in other words, this enzyme exists in the phosphorylated and dephosphorylated state. Now you tell me before I give you the answer. I'd rather have you figure it out first, because you can figure this out. You can figure this out with the phosphate group on. Do you think the enzyme is active or is it inactive? What is your mind telling you? Is the enzyme with the phosphate group on, is it active or is it inactive? And the answer is, now if you did not know the answer, how would you figure it out? Well, you would figure this out by saying, how does epinephrine work? Remember, epinephrine works by phosphorylation mechanisms. Remember all that? Protein kinase A bins. Remember, hopefully you tell me you remember all that. So epinephrine works by phosphorylation mechanisms, right? So the question you ask yourself is, do you want to break down triglycerides when you secrete epinephrine? And the answer is obviously yes. If you're secreting epinephrine, you must be exercising. You need energy. You want to break down triglycerides. And how does epinephrine work? By phosphorylation mechanism. So sure enough, with the phosphate group on, it's active. It just makes sense. There's no mystery here. I told you earlier, way back a few days ago, I said, make biochemistry your friend. Biochemistry wants to help you out. It wants to walk with you, talk with you. It wants to help you out if you just make it its friend. Make it friends with biochemistry. Don't kick it aside. Just know a little bit of biochemistry knowledge. It goes a long way. Whereas insulin, it says here, insulin will shut down the enzyme. Insulin works by dephosphorylation. You remove the phosphate group from HSL, it will not work. So there it is. Just makes sense. How do you think cortisol works? It says here, cortisol turns on the enzyme. How was cortisol turning on HSL? You say to yourself, well, cortisol is a lipid hormone. It's got to work at the gene level. It turns on the gene for HSL, more enzyme, increased velocity, increased breakdown of triglycerides. It all makes sense. Again, connect the dots with endocrinology, physiology, with biochemistry. Just fill in the gap. It all starts to make sense here. All right, now continuing on. Now the glycerol product, the only organ that can deal with blood glycerol gets dumped in the blood. The only organ that can deal with it is the liver, goes back to the liver and picked up the liver, and then the glycerol will then go into gluconeogenesis. Now remember, gluconeogenesis also is turned on at the hormone level. Increased glucagon, increased cortisol turns on gluconeogenesis. Now here, all you got to do is put page 76. Page 76 with this arrow, that's where the details of cortisol and glucagon turning on that pathway of gluconeogenesis here. So the 3 carbons of the glycerol will be used to make glucose here. But remember, we talked about this earlier. The dominant carbon source of to make glucose are the glucogenic amino acids here. Now let me ask you a question. Of the 20 amino acids, I'm looking for a number. What number of amino acids are glucogenic? I know some are both glucogenic and ketogenic. That's true. But how many of 20 amino acids can be used to make glucose? How many? Looking for a number. And the answer is 18. 18 amino acids can be used to make glucose here. And so by default, the easiest way to remember this is, by default, just know the 2 amino acids that are solely ketogenic. And then by default, the other 18 amino acids can be used to make glucose. And the 2 amino acids that are solely ketogenic, again, are the 2 L's. And they are leucine and lysine. Leucine and lysine are solely ketogenic. Here. By the way, another helpful issue here. Remember the two essential fatty acids are the two L's, linoleic, linolenic acid. And similarly here, the two amino acids that are ketogenic help you remember the two L's, lysine and leucine, can only be used to make ketone bodies. And then by default, 18 other amino acids can be used to make glucose here. All right, now what happens to the fatty acids? The fatty acids from the adipocytes get picked up by albumin, and many of them delivered to the liver. Yes, the fatty acids go into other organs, yes, the muscle and so on, but many of them are delivered to the liver. Now, in the liver, as well as all cells, the fatty acids then could go into a major pathway there called beta oxidation, coming up on the next slide. And in beta oxidation, that's where you generate ATP because the beta carbon will be oxidized, and by doing so, generate multiple acetyl-CoA. So beta oxidation generates lots of ATP and lots of acetyl-CoA. Now, if I were to ask you, is there any relationship between beta oxidation pathway and gluconeogenesis pathway? Again, it's not the carbon. The carbon of fatty acid cannot be used to make glucose. But is there any relationship between the two pathways? Up and down with your head, up and down with the head. Because remember, in gluconeogenesis, you're making something, you're biosynthesizing something called glucose. Any pathway is any pathway which is biosynthetic requires energy. So in the liver, it's the energy, not the carbon, it's the energy of beta oxidation of the fatty acid that powers the pathway of gluconeogenesis here. So it's the energy of the fatty acid then used to make glucose in gluconeogenesis. Not the carbon, it's the fatty acid. Now, if you think that's, what's the big deal? Let me show you how big deal this is. Without looking at the next page, because the next page is the pathway of beta oxidation. After we do the biochemistry of the next page, then we get to the pathology. So if a patient, if a child, if an infant sleeping in bed in a crib, 6-month-old child, for example, sleeping in a crib, if at 3 a.m. in the morning, child hasn't eaten anything for a while, if they have a defect in beta oxidation pathway, what do you think is going to be the number one hallmark of the pathology? What's going to be the number one hallmark of the pathology? If they have a defect in beta oxidation, what's going to be the hallmark of the pathology? That's right. Severe hypoglycemia and ultimately sudden infant death syndrome. That's why the beta oxidation defect diseases are associated with sudden infant death syndrome. A child sleeping in bed, 3 a.m. in the morning, the child needs to make glucose, the child needs to make ketone bodies. And where is the energy coming from? Fatty acid beta oxidation. And so if there's something wrong with beta oxidation, the child cannot make glucose, they cannot make ketone bodies, and eventually the child will succumb right there in the crib. Major stuff for the boards. Major stuff here. All right, now in most cells in the body, the end product of acetyl-CoA will go into the citric acid cycle, generating a lot more ATP. But in the liver and liver only, as we'll talk about tomorrow, the acetyl-CoA will go into another pathway, and that's ketone body synthesis, ketogenesis, where you make the ketone bodies by the liver only. They get dumped in the blood and then go to other cells to be metabolized here. All right, now that's what's coming up next is beta oxidation. Then after that, ketone body stuff in this chapter here. All right, now this brings up this issue of the Atkins diet and the ketogenic diet. All right, now what's that about is according to the Atkins people, according to the ketogenic people or whatever, in the human body there is one bad player. There is one bad player in metabolism in the human body. So who is this bad player in metabolism in the human body? Who is the evildoer that the Atkins people are fingering as the evildoer? And who is that evildoer indicated by the blue pointer? Who is the evildoer? It's insulin. They're saying insulin is the evildoer of metabolism. Why? Because when there's insulin around, insulin will result in dephosphorylation of the hormone sensitive lipase. When the enzyme is dephosphorylated, it's inactive and you won't break down triglycerides. So what the Atkins people say is keep insulin low. If you keep insulin low, the phosphate remains on HSL, and therefore it's always active. You'll break down triglycerides, and you will lose weight. Keep insulin low, the phosphate remains on the enzyme, and you'll lose weight. What is the only way you can keep insulin low? What is the only way you can keep insulin low? No carbohydrates in the diet, right? So that's how the diet first started out. They start out as a high-fat diet because you cannot get enough calories from the triglycerides from the adipocyte. So you get calories from fat in the diet, fat in the adipocytes. So the diet started out as a high-fat, no-carb diet. Here, high-fat, no-carb diet is where the diet started out. But then they found out way back 10 to 15 years ago, when the diet started out as a high-fat, no-carb diet, they found out that those people were losing a lot of muscle mass. And why were they losing muscle mass? Because they didn't know their biochemistry. They forgot one thing. What did they forget? They forgot the body needs glucose. Remember that? And so, remember the erythrocyte business? So the body needs glucose. So why were they losing muscle mass? Because of this part right here. They were breaking down muscle protein to generate the glucogenic amino acids to get carbon to make glucose. So once they realized that, it didn't take very long. Maybe they took a Kaplan course, they figured that out. But in any case, they figured that out and say, well, okay, let's change the diet to where it is today. A high fat, high protein, no carbohydrate diet here. So now there's variations of that, whether it's the South Beach diet or the Mediterranean diet. It's all basically the same. It's a high fat, high protein, no or low carbohydrate diet, just variations of the same thing. It's better to use protein from a piece of chicken than your own protein to get the carbon to make glucose. So a high fat, high protein, no carbohydrate diet is where it is today, or variations thereof. Now, is that a good diet or not? There's no doubt about this, people lose weight, but is there a good diet, or is that a good diet or not? Now, my opinion, for whatever it's worth, backed up by nutritional experts, that is a dangerous diet in the long term. Short term, no problem, but a long term, it is a dangerous diet because there are two potential dangers to that type of a diet. Now, and by the way, there is evidence that a ketogenic diet is useful for treating certain epilepsy. So you might talk about this in your other courses. So it is useful for treating certain epilepsies. But for the vast majority of people, in my opinion, for whatever it's worth, it is a dangerous diet backed up by nutritional experts. Now, why is that? And one reason is because of this arrow right here. The glucogenic amino acids to generate the glucose. As we'll talk about tomorrow, what happens is if you're going to take amino acids to make glucose, you got to remove the amino group. That's the first thing the body always does, is remove the amino group. The carbon skeleton is used to make glucose. But now the amino group, you got to dispose of it because it has no energy via the kidney into the urine. So now the kidney is working overtime. It's working overtime disposing of nitrogen, either in the form of ammonia, ammonium ion, or urea, or whatever. It's working overtime disposing of nitrogen. In the short term, no problem. But is the kidney meant to work months and months working overtime disposing of nitrogen, excess nitrogen? I'd be very nervous about that here. And even the Atkins people acknowledge that. If you read their literature, they say anybody 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 on the who has renal problems should not be on the Atkins diet, because the kidney is working overtime disposing of nitrogen coming from those glucogenic amino acids here. So that's one potential danger. Again, a few weeks, no big deal. But the other, long term, a year, I'll be nervous about that. But the second potential danger of the diet is this thing right in the middle of the slide. A person is in perpetual ketosis. Is the body meant to be in ketosis for months and months and months? I'd be very nervous about that. Tomorrow, once we talk about ketone bodies, then we'll get to ketosis and pathology. Is that a problem? Oh yeah, up and down with that head. So again, is your body meant to be in ketosis? I'd be very nervous about that. Now, let me just say something here. Yes, those people on the Atkins diet, ketogenic diet, they are losing weight. But why are they losing weight here? Because it's a mental game. They're losing weight because they're watching what they eat. They're looking around, watching what they eat, so they know don't eat anything with carbohydrate in it. Don't eat all those sweets and so on. Hey. And that's why they lose weight. I'm guilty too. I mean, you would lose the same amount of weight as somebody on the Atkins diet by just watching what you eat. You know, I'm guilty too. If there's a piece of cake in front of me right now or a donut right in front of me right now, and if it's free, I'm going to eat it, right? I mean, mama didn't raise no fool. So I'm going to eat this donut or cake in front of me because it's free and it's tasty. And I got a sweet tooth too. But the idea is that's why Atkins people are losing weight. They're watching what they eat. And you and I would be the same as them by watching what you eat. That's why they're losing weight here. And by the way, it doesn't have to be anything sweet. It doesn't have, by the way, if you ever go to McDonald's or whatever, and for 37 cents extra or whatever, you can supersize the Coke and supersize the French fries and so on. That's, you know how much sugar is in one can of Coke? 6 tablespoons of sugar are in one can of Coke. And if you supersize that thing, you can imagine how much sugar is coming in the body, how much insulin is being secreted in response to all that. But that's why they're losing weight. They're watching what they eat. But it doesn't have to be anything sweet as well. Next time, for example, you go to the movies, and when you go to the movies, just do some people watching. Watch the people when they come into the movie theater. They'll go to the counter and they'll buy a big vat of popcorn and a big Coke or whatever, and then they go sit down and they're watching previews before the movie starts. They're watching all these previews. What do you see? When you do people watching, you never see a person take that big vat of popcorn and eat one popcorn, right? You never see that. What do you see? You see a machine where the hands are going like this, like that, right? Just into the mouth. And then before the movie even starts, they go back to the counter and get another big vat of popcorn. And popcorn's not sweet, yet it's all carbohydrate. Yeah. So the idea is that's why they're losing weight. They're watching what they eat, these Atkins people. And you would be the same as them, and I would be the same as them by just watching what you eat. Don't eat between meals. Watch today how often you and I, especially me, how often we reach for something sweet. And if you avoid that, you're going to lose weight. So that's why it's truly a mental game here. Okay, now I'm looking at the time. Perfect time. It's at the bottom of the hour. Now tomorrow, when we come in, again, we got 4 hours, and every slide high yield. I'll make it worth your while. And again, don't read ahead. We're going to skip over some things that are low yield, but all the figures we're going to do are going to be really, really high yield stuff. Lot of stuff coming your way tomorrow. And so again, today I'm going to post, I think, 3 more questions on the Facebook today. and then 4 tomorrow, but 3 today. I'm going to post. I'm going to put the answers to the previous 2 questions I did yesterday, and I'll post 3 more additional questions based on all that cholesterol stuff we did today as well. And then, and so we're right where I want to be. And by the way, more students are looking at Facebook. Right now, we're up to about 20, 25 students are now looking and posting questions, answers on Facebook. Right. But again, I'm looking for hundreds of students, but only 20, 25. But I'll take it. It's better than nothing. Okay. And then I want to thank Dr. Fadi, Dr. Mir, and Dr. Soto again for been with us all week. And I want to thank them so much for content assistance. And obviously always thank Maggie for wonderful technical assistance. She always has my back, you know, and when I forgot to turn off the microphone, she know that guy Turco doesn't know what he's doing, shuts off my microphone. So thank you, Maggie, for doing that. Okay, everybody, thank you so much for helping out. And the students, thank you so much for showing up. One more day to go. It's hard to believe. We're almost done. One more day. You know, after I get to know you guys and, you know, and so on, then we got to leave tomorrow. Anyway, it's kind of sad after, you know, all this time. But in any case, I'm looking forward to seeing you tomorrow. Have a great day on Friday here, and see you tomorrow promptly at 9:30 Eastern time. So take care, and God bless you all.
you.